Hello, hello. Start next special lecture. Okay, good afternoon, everybody. Hope you had enjoyed good food. Yes or no? Okay. So now we have with us a renowned taxonomist, and I find myself fortunate enough to introduce her because I worked in her lab for some time. During my PhD, part of my work I conducted in her lab. So I am very happy to introduce her, Professor Sasikala, former professor, Center for Environment, Jawaharlal Nehru Technological University, Hyderabad. Her research area are bacterial discovery, metabolomics, biodegradation, and bioprospecting. In her 30 years of research experience, Dr. Sasikala have published over 200 research papers. She has received many prestigious awards, including DST Young Scientist Award, UGC Research Scientist Award, DBT Overseas Associateship Award, UGC Research Award, UGC Mid-Career Award, and she is also recipient of very prestigious AMI Professor B.N. Johari Award for Microbial Diversity in 2016. Dr. Sasikala is also elected member of International Committee on Systematics of Prokaryotes, Subcommittee on the Taxonomy of Phototrophic Bacteria, and New York Academy of Science. Dr. Sasikala was nominated as an expert to Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services of United Nations by Ministry of Environment, Forest and Climate Change, Government of India. And she has also worked as a resource person for evaluation of DBT, DST, MOES, and MOEF projects as well. She has guided over 26 PhD students and 3 postdoctoral fellows. Dr. Sasikala has completed over 19 major research projects and have 4 patents. Professor Sasikala has identified over 136 species, 18 genera, 1 family, and 1 order. She has developed novel methods for isolation and enumeration of anaerobic bacteria, selective enrichment of green sulfur bacteria, and rapid and inexpensive typing method based on FTIR spectroscopy. Dr. Sasikala applied new methods like MLSA, catabolite barcoding, and metabolome analysis for taxa description. So I humbly request her to please come on stage and deliver special address on bacterial diversity and bioprospecting. Please, madam. Next, please. And this is
Pointer, yes.
pointer. Okay. Yes.
Good afternoon, one and all. First of all, I thank the organizers and of course Dr. Hitarth for giving me this opportunity to face this very young and active and exuberant audience over here. Only thing is it is a post-lunch session. So it is a challenge for me to keep you awake. Let me see how much successful I'll be in that. It was a great. Okay. Okay. Is it audible now? Okay. So I'm experiencing great hospitality of Atmiya University, enjoying, I mean, it is not today. From the time when I was invited itself, so meticulously everything is being planned, and I got a call yesterday at 11 o'clock, madam, your flight is there at so and so time, like that. Everything is being followed up, and thanks a lot for Dr. Aksha and Dr. Hitarth and other organizers of the conference. Okay. I think in the morning also you heard a lot about bacterial diversity. Probably it is a part of whole bacteria, a specific phylum, which was earlier called as Actinobacteria. There is a new name given to it. Let me see who will find it out. What is the new name of the phylum Actinobacteria by the time I complete my talk? Okay, that is a task for you. I'm going to speak in terms of for environmental. Okay. Can anybody identify or say what it could be? Why this design on a scientific presentation? Something like a rangoli? I'm from South. Every day we put rangoli in front of the house. Why this? Can anybody predict? These are bacteria. These are bacteria. Every one of them is a bacterium. This is a Petri plate on which the design was made about 15-20 years back, and from then on, every single slide presentation of me has this as the cover design. So we are going to see. So whenever I say environmental management, people think that it is only wastewater treatment. It is not so. Whenever you are trying to reduce the production of pollution, that is much more important than treating the pollution after pollution has taken place. So this will be prevention, where you are going to use eco-friendly processes and bioproducts. And believe me, today everybody is speaking about biotechnology, nature-based solutions for everything bio, and microbiology is coming into picture. And it has a great entrepreneurial capability. You can be a very good entrepreneur and have very good business. Isolating and marketing microorganisms. There are several companies which do it. So whatever you are spending time here for your PGA or UG learning microbiology, you can always have that on the back of your mind, and you can start your own venture growing bacteria. I'm coming to it much later then. And then whenever we say treatment of waste, so if you are doing through aerobic process, what are the products? Aerobic degradation of organic matter will lead to the production of what gases? carbon dioxide. And what is carbon dioxide? It is a greenhouse gas, global warming. But never forget that carbon dioxide is also a plant nutrient. It is a nutrient for this only because the concentration has increased today, it is a problem for us. Otherwise, carbon dioxide is good only. It's not bad. Because the concentration is more, it is a global warming causing. So when you are doing it, you are contributing to, you are thinking that you are actually treating waste, but you are contributing to global warming. So it is better you convert the waste into wealth. So let us see how we are going to do it. So working on bacterial diversity is an opportunity for you to go all over India at least on duty. So you may spend your own money if you are very passionate about it, but it will be on duty. And like this, we have gone to Pangong, which is the northernmost thing. This is the lake which is shared between China and India, Rameshwaram, Kutch, and then Chirapunji. So much we have roamed about on duty. My husband also works in the area. So as a family, we all travel a lot. And these are the various environments from which we have isolated, and many isolates from Gujarat also. For 3 years, I had a project to work on marine microbiology, marine bacterial diversity of Gujarat. So it is like, there is nothing like you can isolate bacteria only from here and not from there, like God, omnipresent. Bacteria are omnipresent. You go to temperatures as high as 150, 200 degrees Celsius, you find bacteria? Is it possible? Where? Okay. And then you go to sub-zero temperatures, there is life. And you go to 12 pH, you go to 2 pH, there is life. And it is mostly bacteria. And then these are the various places from which we have isolated bacteria, all colors. You can see this is a cold sulfur spring in Gulmarg. The water was coming out there, and I found, so this color attracts, and this is a brown pond near Rameshwaram, and you can see that there is actually these coconut trees, and you can see the reflection. And we went there. The moment I saw this, I know these are anoxygenic phototrophic bacteria, the Rhodobacter, Rhodosudomonas kind of organisms. And then we started collecting samples there, and the local people, they came, they asked, why are you collecting samples? We told, like, I mean, you feel, no, some unpad person has come and you are a scientist, you are going to say, so there are bacteria in this because of that. And you should see the type of smile that was there. It told, you, person without common sense, actually, we are soaking those leaves of the coconut tree there to take broomstick out of it. So the color is because of that, they told. But luckily for us, we did find a new rhodovulum species there, which is named as Pheolacus. Pheo is brown, lacus is lake. And then this is Rhodobacter chanclensis on Changla Pass. And this brown color, again, we thought it is our bacteria, like photosynthetic bacteria. And yes, we found a new species here, but this brown color actually is not because of that. If you remember, if you have studied in your intermediate, that is +2 or so, there is one alga, green alga, which has this color. Chlorella species. So that forms on ice. That's known to form like that on ice. And this is Rameshwaram. And even Kanyakumari, you have this colored sand. They sell the sand also, colored sand. And it is because of a new species that we have described, Littori. And then this is a jellyfish. You can see these colors, you know. So they are, Dr. Dayanand Aksar was telling that color is because of minerals. That's what even geologists say. When I went and when I say bacteria, they say, "Arey, you don't know anything." Color of rocks comes from minerals that are there in the rock. But it is not always so. It is possible for bacteria to give colors to rocks, sand, etc. And this is a pink pond. This is actually a picture from Hindu. So the moment we saw, we knew it must be a photosynthetic bacterium. You take it. It looks like the culture that we are growing in the lab, and you centrifuge, observe under the microscope. This fellow is responsible for the color of this particular uh, uh, pond. And then you have this purple pond near Satpada where uh, uh, what prawn was being washed. And here the pH is more and you get this particular bacterium growing in abundance. And now, after I have explained all these things, you may think you go and find a colored body. So you are likely to get a photosynthetic bacterium. Here is this pink pond Bhavnagar near Nari, and it was so beautiful to observe this. During different times of the day, the color ranges from pink, purple, orange, like that. These bacteria are both photosynthetic and anaerobic. So depending on the day, they have to adjust themselves at a place where light is available and there is no oxygen available. So they keep moving, that's why the colors will change. But then you can see that we could not actually grow that purple bacterium in the lab at all. When you do metagenome analysis, you see that 70% of it is this thiocapsa, which is so abundantly growing in the nature, is simply not growing in the at the lab at all. We even centrifuged the same sample and added some nutrients and tried to grow it. It refused to grow. So that is for bacteria. There is what is called as great plate count anomaly. When you observe under the microscope, you can count the number of cells that are there. You do the plating, you may get only 1% of the bacteria growing on the plate. That is called as great plate count anomaly. That is why there is so much importance being given to metagenome analysis, where you isolate the genomic DNA and then sequence. Then from the sequence itself, you will come to know what is the quality, what different species are present, and then what is the relative abundance of them. So that is the this thing. So this is the actually culturing the characterization of the bacteria for description of new species. Dr. Dayanand was explaining in the morning. And then you have to do all ecological. You have to know what are the at least temperature pH of the place from which you are isolating the bacterium. Morphological, both on the plate, culture, and then under the microscope. Physiological, pH, temperature, etc. And then biochemical characterization. Chemotaxonomic is the very challenging one, like how he was telling. And then genetic characterization. And of course, today, sir was only speaking about 16S rRNA gene sequence, which is just basic. Today, doing genome sequencing has become compulsory. But one good thing is the cost has come down tremendously. When we first sequenced the genome in 2005, it has costed us 6.5 lakhs for one bacterial genome. And today it is just 20,000 rupees. Just 20,000 because comparing to 6.5 lakhs. This is compulsory. And when you go on seeing, when I started doing description of species, the amount of work that was being done was very limited. Now it must be at least some 10 times more than that. And a lot of work need to be done with this phylotaxogenomics and then predicting various characteristics from the genome analysis. Lot of bioinformatic work also is involved. So taxonomy is no more boring. So it has molecular biology or bioinformatics. I think that is very glamorous for the students. So whenever you ask, that's what they say. So whenever you are doing these chemotaxonomic characteristics, okay, I think we are with a different thing. Okay, whenever you are doing, you are also using a lot of characterization methods. For quinone, you will have to do HPLC, fatty acid composition, GCMS, polar lipid composition. Again, you can do 2D TLC, or you can go to GCMS, carotenoid, again HPLC, hopanoids, GCMS. So you are using different methods. And this is compulsory for us. We have to do characterization, which is a basic fundamental work. And I am with a university called as Jawaharlal Nehru Technological University. And people used to ask, how do you justify my presence in a technological university doing very fundamental taxonomy work? Doing taxonomy work, we know whether a particular bacterium is capable of producing all these things. And that can be exploited further. Yes, this is what different methods that are being used as listed here. So this is the total number of various taxa that we have described based on cultured diversity. That is diversity of bacteria which we have cultured. This is the work of two different laboratories, mine and my husband's together at University of Hyderabad. So we work together. Uh, I think in India, ours is the leading group. It is not a small thing. But now, I told you, no, because most of the bacteria cannot be actually grown in the lab. And today we have the capability to sequence the genomic DNA and then see what is there. And today it is possible for us to get whole genomes from the metagenome. So from that you can describe new species, that is metagenome assembled genomes. And then you can give new names and describe new species based on metagenome assembled genomes. There is another one called as single cell assembled genomes or SAGs. So for students here, you may think that if you have to do business on microbiology, you have to have a sophisticated lab or you have to have a lot of knowledge, nothing of that sort. There is lot of demand for biofertilizers. And biofertilizers, which can be nitrogenous activity, of course, for this you would require a GC. It can be phosphate solubilizing, it can be potassium solubilizing, or it can be zinc solubilizing bacteria. What you need to do is just to do a plating in which you will add the insoluble compound, tricalcium phosphate for phosphate solubilization. And after you inoculate, when the growth comes, if it is solubilizing, there will be a zone of clearing around it. That is enough. And there are people ready to buy the cultures from you. 20,000, 30,000 for each culture. It can be a simple classroom experiment which can give you very good solubilizing bacteria. So if you want, somebody is asking for some bacteria to be used in a particular area, you can take soil from there and isolate. And you know, when it is put back into the field, it will definitely grow in that area. If you are taking something from Hyderabad culture and you are growing in, trying to grow here, the climatic conditions are different. It may or may not grow. If you are isolating from the same place, it is already adjusted, it will definitely grow. It's a simple experiment you can do. Antimicrobial activity, I think it will be a part of your classroom experiments you can do. Indole 3, of course, for this you will require GC. So just to show that paddy root is inhabited with lot of purple sulfur bacteria, and a few of our bacteria are commercialized and they are being used. And biofuel, I'll not go much into it. Photosynthetic bacteria can be for hydrogen production. This was my PhD problem. I worked, but then itself I know it does produce hydrogen, but the production rates are very limited. So it is not commercially viable. Even today, it is not commercially viable. And they produce a lot of fatty acids. So this is one organism, the first organism I had worked with, isolated from Osmania University, and we have demonstrated hydrogen production from wastewaters also. And this culture is being used by several countries, several people have took this culture from American ATCC, American Type Culture Collection Center. And that is a story, you know, when I sent a paper for publication, that paper came back, rejected, saying that probably there is a contamination in the culture. Just then I had received this certificate of deposition. Then I wrote back saying that it is deposited and they have confirmed that it is Rhodobacter spheroides and it is a pure culture, and it was published. So whenever your papers get rejected, don't be afraid or anything. Attend to the questions that they have asked, and it can be, you can submit it again to the same journal and it can get published. Fatty acids, whenever there is fatty acid, that can be used for biodiesel production and also as a nutraceutical. It has medicinal value. I'm not going into details of it. So there are bacteria which are having these polyunsaturated fatty acids, long chain unsaturated fatty acids with very good percentage of this, but we have not worked on biodiesel production. If somebody wants, I can give the cultures to you and you can work on that. We were interested in taxonomy and went on that. So these also have good amount of lipid content, which makes them oleaginous, more than 20. And then they are, you have seen the bacteria, the photosynthetic bacteria that we had been working with. One challenge with photosynthetic bacteria is they are anaerobic. Anaerobic bacteria, not many groups are working. And we then started working on other anaerobic bacteria like Clostridia, spirochetes, sulfate reducing bacteria, because of which we could get many, many new taxa and describe such large number of new bacteria. So these can be simply coloring substances. You know, all those synthetic dyes that we use are carcinogenic, blah, 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 all those things. So you can use this. And of course, apart from that, it has so this is a patent that is already granted. So from the test tube, you can do and actually grow them. You don't require any bioreactors or anything. You can just take a 20-liter container and grow, because there is no need of any aeration, anaerobic bacteria. Because of that, the cost of production is coming down. Always giving aeration is a great energy consuming activity in culturing the microorganisms. But light you have to provide. You can just put it outside also. Only night there will not be light, so growth will be less. And when there is light, there will be good growth. So you can centrifuge and you can actually extract using a solvent extraction. And we could get actually highly pure compound compared to whatever is available with Sigma Aldrich 99%, and the cost also is very, very minimal compared to that. So here is another pigment, green color pigment called as neurosporin. Two different bacteria could do this. And now we are growing this bacterium not on any medium that is prepared, but on wastewaters. So you are achieving both the things. I'll come to it a little later on. And this is one interesting story, because there was one company in Hyderabad which got an order for mass cultivation of Rhodospirillum rubrum, which is a purple non-sulfur bacterium, to supply to a European company who want to use it as a to poultry to reduce the cholesterol content in their poultry, in their eggs, especially in their eggs. And they went to different places, including MTCC and other things, and somebody directed them to my lab. I mean, this is the work that we are doing every day, photosynthetic bacteria. It is a challenge for others because they do not know how to cultivate anaerobic bacteria, which is just child's play for us. So whenever you are selecting a topic for your research, if you select a topic which is not really worked by many people, the chances are better for you to get recognition. And another thing is, we have to accept that there are challenges for us as far as facilities are concerned in India. So I've shown that you require so many facilities for doing it. So if you are working on cancer biology, or if you are working on other things where the developed world is working, we cannot compete with them because we do not have such type of resources at all. But India is a mega diverse country, and we have lot of biological resources, and bacteria just unending. So if you work on bacterial diversity then you will become a world leader, or you will be well known in the world. Like I have become a member of International Committee on Systematics of Phototrophic Bacteria, because there are not many, because India is having rich resource of biological resources, and that I can always tap on. If I have to work on cancer biology, the type of or an Alzheimer's or other things, I cannot match them on the resources. That's it. So this is, they came to me, and then we have mass cultivated, and we got a project from that, out of which I could buy small instruments. So one of the students, he was there, so he wanted to try if our bacteria also has hypolipidemic effect. The experiment was, you take rats, two sets of rat. One set is having high fat diet. The other set is having we are providing it high-fat diet along with 2% of this particular bacterium called as Blastochlorus gulmergensis. And after some time, you will have to sacrifice, you take the blood sample and you estimate the uh, lipid content that is called as lipid profile. Lipid profile contains total cholesterol, triglycerides, total cholesterol, triglycerides, high density lipoprotein or HDL cholesterol, and LDL cholesterol. So you can compare. Blue is only on high fat diet, red is high fat diet plus the bacterium. You can see there is less cholesterol and triglycerides, which is actually the one which causes atherosclerosis and all those things, that is significantly reduced. And LDL cholesterol also is very significantly reduced. They say, for triglycerides, they see in terms of 150, 200, 250. For LDL cholesterol, 100, 101, 102, 103. So much bad effect this LDL cholesterol has. But HDL cholesterol remained the same. So what to do? And HDL cholesterol is actually good cholesterol. So you have to have HDL cholesterol should be more. And luckily for us, our bacterium has not reduced HDL cholesterol, but reduced all the other cholesterols. We did further work and we have seen what is the mechanism of action, what enzymes are inhibited, why is this happening, that also we have seen. And this is one thing. I'm with Center for Environment, so naturally I'll have to work on waste degradation, etc. We started working on especially the N-heterocyclic aromatic compounds, I told you, in waste treatment. But then whenever you're doing that, it is actually a loss of resources and it can lead to global warming. So we have to think of waste valorization. And then a concept called as circular economy. Whatever comes into your campus, let us say Atmiya University, nothing should go out. If you are using water, you have to find ways of treating that and then reusing probably for your garden or something like that. So in that concept, so we have worked on indoles and a lot of products of indole were produced by our bacteria. You can see that they are all, I have not shown anything here. What we actually do is grow the bacterium on the medium containing it. Then you will have to centrifuge, remove the bacterium, and then you will have to extract the compounds, and then you will have to purify it, and then you have to identify the structure based on what is called as polychem analysis, where you will do NMR, you will do GCMS, LCMS, and you will do NMR, FTIR for functional group, NMR for carbon, and these things, and you can elucidate the structure. Like that, different compounds were, including melanin also, of course, and then they were all bioprospected for various biological activities, but we have never used it. This is an example. So you can see that this is an indole moiety, and this is a carotenoid. So it is a high molecular weight indole ester. And we all know that indole 3-acetic acid is IAA is a plant growth promoting hormone. So this also may be having plant growth promoting character. You can see this is mulberry tissue culture seedlings. So they have done all this optimization, and with that NAA and 2,4-D, they could get only this much of rooting. Whereas with the purified compound rhodestrin, it was better, but with just acidified extract. You grow the culture on indole, centrifuge, and you take the supernatant, acidify it, and then use. It has very good rooting. Of course, if rooting is good, it is able to get more nutrition, so the plant will grow better. And this is one area where commercialization also had been done. So photosynthetic bacteria are purple sulfur bacteria and some non-sulfur bacteria are capable of degrading hydrogen sulfide. In aquaculture ponds, you give feed to the fish. So some part of it will go back into the sediment. What happens when there is degradation? Oxygen is utilized, so there is production of anaerobic conditions. And during these anaerobic conditions, if degradation is further going on, there will be production of hydrogen sulfide, and ammonia also is released, and these are toxic. I don't know how popular is aquaculture here, because it is all, you have a large coastal area. Is aquaculture very popular here? No. AP is very popular, and then because they use a lot of chemicals into it to take care of the vibrio and other things. And whenever they are laden with antibiotics or other insecticides, pesticides, etc., that cannot be exported, and they get money only when they export, not in the Indian market. So now lot of probiotics are being used in aquaculture, and it is already known, and our bacteria have given to the industry. It was just a newspaper article where one person saw it. He was only a consultant, where he was suggesting products for aquaculture people, and they were importing bacteria from, I mean, something like a medicine which has bacteria from China and giving. Now that person has established his own industry where he is making these products for taking care of ammonia, and then for this thing, and especially poultry waste. For poultry litter, you know how bad smell it comes, of course. Yes, I was asking you for aquaculture. I shouldn't ask that question because it's predominantly vegetarian society. So what will you do growing all these things? There is no reason. Yes, that's why I was surprised. I was thinking why there is no aquaculture with so much coastal region in this thing. The other one is oil bioremediation. And whenever we speak of petroleum, diesel, or crude oil bioremediation, the main problem with that is, can you think why we do speak so much about oil pollution, oil remediation? Why is it so difficult to take care of oil when oil gets into water? When petroleum, diesel, or crude oil gets into water? Simple, it is not miscible with water. And you know, the basis of life is water. Wherever there is water, there only life will be there. If it is water vapor, no life. If it is ice, no life. So now we'll go back to our question. Why is there life at 150 degrees Celsius? Why is there below 0 degrees Celsius? Two statements I'm doing. One, if life has to be there, water should be, H2O must be present as water, not as water vapor, not as this. And then, but you find life at 150 degrees Celsius, bacteria growing. You find life at -4 degrees, bacteria are growing. How is it possible? So what was the first question? What is the new name of actinobacteria? So be thinking about this. Probably also listening to what I'm uttering here. So this is one organism. The importance of this bacterium is it is capable of producing biosurfactant. So if you are adding something to make the oil miscible with water, it will get biodegraded. So along with bacteria which can degrade diesel, you also should have a biosurfactant producer for bioremediation to take place. We made a consortium with these bacteria which are capable of degrading diesel, and also this is a biosurfactant producer, and you can see this is before and this is after the concentration of various components of diesel. It is not one thing, a number of hydrocarbons are there. They have reduced significantly, and this has been used actually in Secunderabad railway station, wherever they actually what do you say, fuel, add the fuel to the train, there will be some spillage, and it has become a very thick layer. And whatever they are doing, they were unable to get rid of it. They want to get rid of it not because of aesthetic reasons. If it is dark or if it is dirty, it is okay. But it is a fire hazard. So they wanted to get rid of it. And the company I told, which I helped them to establish technically, so that company made this thing, and then they have, it is again commercialized and it is in use. And this is, but whatever these what ocean and seas, wherever you see, it is mainly crude oil crude oil contamination that happens. It is because crude oil is transported most of the time, and then refining takes place, and it is mostly transported through the land. That's what India also is doing. So whenever there is marine, it will be crude oil bioremediation. So what we did was, we got crude oil from, I just put a mail to two different petroleum university and some other university who are working, and they just sent me the crude oil samples. And then we isolated all this bacteria from that, and all the strains, we have confirmed that they are capable of crude oil, and we have done what is called as soil microcosm study. The earlier diesel also is a soil microcosm study. This also is a soil microcosm study. So here we have used what is first, only nutrients are added, or here you add only the biostimulation is adding only the nutrients, and this is adding the microorganism. And another treatment was adding both the nutrients as well as microorganisms to it. Let us see what happens to the result. So when you see here, this is sterile soil. When you see, this is control where we have not added anything. We are only looking at the microbial growth, that's all. This is biostimulation, wherein you have added the nutrients. So it is, there is good growth, but the degradation is not much. This is degradation, this is microbial number. When you don't add anything, the number is remaining like that. There is still little degradation. And when you are adding nutrients, there is more microorganisms and there is enhanced degradation also. And when you are using augmentation, that is when you are adding the microorganism, because we have added, of course, microbial number is more and the degradation is very much, though the number is less than that of biostimulation, the degradation is more, simply because here whatever bacteria are there, they are degrading bacteria. Here the bacteria that are there, only small percentage of them are degrading, others are not degrading. And when you add both bioaugmentation and biostimulation, that is, you add both bacteria and nutrients, and you get very high degradation rate, bioremediation. So within 20 days, we are able to get this degraded in soil microcosm studies. And another important thing was, you can see when you do it in sterile soil, when we have to use it in field, actually, we cannot sterilize all the contaminated soil and then do the experiment. It is just not practical at all. So you will have to use only unsterile soil. So you can see that unsterile soil with bioaugmentation, there is very good degradation that is happening, more than what you find with sterile soil. So with sterile soil, it is this much, 100, it is 95 or so. And with unsterile soil, it is almost 100% bioremediation or biodegradation is possible. And this also can be commercialized. Now we are trying to take a patent and then give this thing. And all of things that we have seen till now is only regarding, whenever we are characterizing bacteria, there are so many things, GCMS, HPLC, and all these things are required. You may think that you don't have those facilities here in, I don't know whether you have or not, you may not have. Then you don't worry about it. If you are sure that you are having a new taxa, you can just put a mail to anybody who is working in that area, and you can go and work in their lab and then get the publication. That's what your own Dr. Hitarth Bhatt did for his PhD work. He put a mail, he came and he worked, and he got a, it was a new genus also, Desertibacillus. That's all, no? So that is possible. Whatever you can do in your lab, you do it. And then you contact people who are working in that area, and you may say that you don't have the facilities, and you can say that it will be a combined publication. Sometimes even without that, people do, accept you to work. So you can do that. So for working, facilities are never a problem, as long as you can prove that it is something new you have in your hand. So all these things we are only speaking about organic compound. Bacteria, they are living organisms. They have to only deal with organic compounds, no? Will they do something with inorganic compounds? Will bacteria do something with inorganic compounds? Can they? How? Life is organic. So they take reduced, the students here. Can living organisms deal with inorganic compounds? What? Examples? Nitrites, sulfates, sulfides. All inorganic compounds the plants are taking. Of course, not we. And then they can also produce inorganic compounds. Can we produce inorganic compounds? Can human beings produce inorganic compounds? No? Yes? Don't know. Carbon dioxide. What is carbon dioxide? Are we not producing carbon dioxide? But there are bacteria which can produce inorganic crystals. Sometimes, we don't realize what we already know. So these are inorganic compounds that can be produced by bacteria. So this is calcite or calcium carbonate. Actually, I wanted to ask these questions to Dr. Dayanand Aksar. There are limestone quarries. Probably part of it is generated by microorganisms. Have you ever gone to the coastal region? Your Viraval and other things. Did you find anything peculiar in the sand over there? What is that you have found? You found the sand becoming hard like a rock. Did anybody find it? Observed it? You find sand near the coast, but a little away from it, it will be hard, almost becoming like a sandstone. It may be because of bacteria which are producing calcium carbonate. Calcium carbonate is a binding material which will bind your sand crystals and make it into hard material. So the same thing can be utilized for biomotor or bioconcrete. I teach students of MTech Environmental Management, most of whom are civil engineers. If I have to give them a project work to work with, it has to have some relevance to their basic training in civil engineering. That's why we went into this. It is a very simple test. What you have to do is just take sand into it, pour the culture from the top, and after a week or 10 days, you just lift it. And then you can see that if it is a calcium carbonate producing organism, it will bind and the sand will not fall down. It will stand like a pillar. Very simple experiment. So probably you have a civil engineering department here. Yes. So the microbiology student can isolate and then give this culture to them. They will do add it. You have to mass culture and then give the culture to them. They will add and they will do all shear strength and what all they do. I mean, even a simple BTech lab is enough to do those experiments. And the cement in the construction industry is interested in this because we had publications with civil engineering department with that, and L&T contacted the professor asking for the bacteria. So that is the potential that is there. So these are the mortar blocks that are done. So you can see here, these white precipitates that you find are calcium carbonate. So you can see this can not only, I mean, you can, when you add it during the formation of concrete or this thing, it can be helpful. It will increase the strength. Whenever there are actually cracks, it can result in self-healing. So actually, whenever there are cracks, they use epoxy resins. This is what civil engineers do. And you can actually apply your culture over here, and then it will produce calcium carbonate and seal the thing. Same thing with epoxy resin. Same thing with our bacteria. This is what happens. This is Bacillus subtilis, our bacteria that the civil engineers had used to do the work. And another inorganic crystal that can be produced by microorganisms is called as So ammonium, magnesium, phosphate. So does it give you what could be the importance of this? What are the different elements that are there? N, nitrogen, P, phosphorus. What is importance of N and P? They are plant nutrients. Whenever you are doing cultivation, farming, the major elements that are required are carbon, nitrogen, phosphorus. Carbon is coming from carbon dioxide, and we normally use nitrogen and phosphorus fertilizers. And this crystal is having both nitrogen and phosphorus, and this is slowly soluble in water, so it can give a good continuous supply of this thing, and this can be used as a means of ammonia recovery from probiotic wastewater. This is what an industry had come to me asking for. They want to take care of ammonia. Whenever they treat also, ammonium is remaining. Because of that, they are unable to send the wastewater into the open water bodies. So we have used this bacteria, which is, okay, this is, okay. So you can use this, what is this Selnicoccus? Sorry. This is the one halobacillus, which is forming this magnesium ammonium phosphate. These are the crystals in the medium, and this can be used as a fertilizer from wastewater. And then this is one bacterium which is capable of producing halide crystals or sodium chloride crystals. So probably this can be a way of desalinating water. For Gujarat, probably it may have significance. If somebody wants, I can give the culture, Selnicoccus. You can run the water through a column containing the bacterium, and then you can see if desalination is possible with this. This shows that how the crystal is growing and all the aggregation, and then how the halite is growing. Halobacillus. What is this? Okay, you can, I think you must be having carbon sequestration, carbon mitigation, and all those things. And for that, we think that it is only plants which can do it, but it is not so. You can have it, bacteria can do it at a much faster rate compared to plants. So this is, I was telling, this is a reactor in which it is just growing on wastewater, which is diluted with only mineral salts medium containing magnesium sulfate, KH2PO4, that's all. Nothing more than that. It is growing on that. And then now you can see there is significant reduction in COD, BOD, nitrates and phosphates, not much reduction. And then you can see that you can get very good biomass yield. And this is a nitrogen fixing microorganism. Because it is a nitrogen fixing microorganism, it can be used as a biofertilizer, and it is actually already being used as an aquaculture probiotic. And then we have seen that we had a actually patent for the production of neurosporin already, and dead biomass can be used as a feed supplement, or it can be also used as a coloring agent because of the green color that is there, which is also this. Of course, I was of use, but what happened to the supernatant that is produced? That is again a liquid waste. So if you can use that water for some purpose, it will be good. So what is done for that is, we have done some simple germination experiments, and you can see that until 10%, there is no inhibition at all, and you can use it till 25%. You can dilute this wastewater with fresh water and use it for irrigating your plants. And also there is good amount of phosphates still remaining in it. So this phosphates will be useful for the growth of the plant. And this is one new concept. We all know that antibiotic resistance, in the morning also people were thinking, but never think that antibiotic resistance is found only in contaminated places. In pristine places also, you will find antibiotic resistance. But how? In Antarctica, you go where there are no people at all and take samples. And if there are bacteria there, there will definitely be antimicrobial activity there, antibiotic resistance. There will be antibiotic resistance over there. Why will antibiotic resistance be there in a place which is not contaminated? From where are the antibiotics coming? They are produced by bacteria. So in any place, there will be some bacteria which are producing antibiotics. So if other bacteria have to survive over there, they will have their own mechanisms of coping up, whether not allowing the antibiotic to come inside or degrading the antibiotic, or several various mechanisms will be there. So you will find antibiotic resistance everywhere. So it is becoming more and more difficult, and operation successful, but the patient died. Patient died because of very infection by a multi-drug resistant bacterium. There was some strain, that Delhi something. It gave very bad publicity to India. India is known for medical tourism, because in the West, there is lot of waiting time. In India, it is not so, and very good after operation, post-operative care. You can get 24 hours nurses available at a very reasonable price in India. It is not so in the West. So lot of medical tourism is happening and people are coming to India for operations. But when they know about this multi-drug resistant bacteria that are there and it may lead to death, there will be a problem. So for that, what you can do is, there are certain bacteria which eat other bacteria. They are called as predatory bacteria. Eating means it's not that they are going to engulf and eat, but they produce certain enzymes which will solubilize the bacterium and they take the nutrients and then they survive. So this is a simple experiment that is done. First of all, you have to grow the bacterium as a lawn. Lawn means, it is totally spread over the plate. And after the lawn is grown, now you can use different bacteria. So this is actually a disc, paper disc, which is loaded with bacterial culture. This is only the medium. This is a yellow colored bacterium, and this is a white colored bacterium. And you can see this bacterium that is there is eating away the already grown bacterium, which is a mycobacterium. Mycobacterium also is very difficult to treat. It requires at least 3 to 6 months of continuous taking medicines, and not one. 3-4 medicines have to be taken continuously, and it is very difficult to really stick to that, because probably 1 or 2 months into taking the medicine, your symptoms go away. After that, to have enough motivation to take so many tablets every day for so long is very difficult. That's why multi-drug resistant mycobacterium also is a very major health threat in India. Of course, in the West, because of HIV, which is reducing of course, now it has become a past, but because of that, immunity is going. So there also it is a problem. So you can have these bacteria which are eating away. So if you can give this, that is a live antibiotic. These experiments also can be done in the MSc lab as a part of actually your MSc lab also, practicals also. Simple things which you can do. And when you have something of that sort, if you want to know what are the, how is it doing and all those things, you can always go to any other lab to do. So here you can see yeast also can act as a predator on bacteria. So this is yeast. So yeast cell is actually attracting the bacteria onto it, and after that it is forming a encapsulation where the bacteria are being, because of the enzymes produced, and then now you see, and it is dividing. When it is dividing also, some bacteria are remaining like that. So you can also, and yeast infections also are very difficult to treat. You can use bacteria to do it. And this is one experiment which was actually done by a student who came for Indian Academy Summer Research Fellows. Hope you know for that and you apply for it. No, Indian Academy gives 2 months fellowship, about 12,000 per month, and then train fare it will give to go and work in selected laboratories. So in JNTU, my lab is recognized. They come there. And one by I gave him because India, Hyderabad is known for antibiotic production and also antibiotic resistant containing, AMR, antimicrobial resistant containing microorganisms in the water bodies, surface water bodies there. And ciprofloxacin was one of the antibiotics like that for which lot of antibiotic resistance was reported. So we wanted to see if we can get some antibiotic resistant, some microorganisms which can degrade actually ciprofloxacin. So we prepared a plate with ciprofloxacin as sole carbon source, then inoculated, some colonies have come, purified it, and we started trying to identify it. We wanted to do 16S rRNA gene sequence analysis. But when we were doing this 16S rRNA gene sequence analysis, first time it failed, second time it failed, third time it failed. Then didn't understand because that is a routine experiment that is done in the lab every day. And why are we failing? And then we observed under the microscope, and this is what we found. They are actually yeast cells. You can't really distinguish yeast colonies from bacterial colonies so much. Yeast is colored always, but we were anyhow working with colored bacteria. It may be a little more shiny than bacterial colonies, but not necessarily. In a plate, if yeast and bacteria are there together, only by looking at it, we cannot distinguish. And it is not just that. Can you see anything else also here, apart from yeast cells? And these inside ones, of course, this is not a video. There was something present inside which was moving, which was actually moving. And the moment I found it, I thought these must be bacteria. And then to our surprise, yes, they were bacteria. So we did metagenome analysis and all that. And then the same thing, Dr. G.S. Prasad, who was with MTCC before, is with University of Hyderabad now. And he is an expert on yeast. Then we said him, like this, we found bacteria. Then he thought, I also found them something moving inside, but it never occurred to me that they could be bacteria. That is the importance of observation. That's what Dr. Dayanand Aksar was also telling, you know. So you find this, they are bacteria, and this led to actually 2 PhDs after that. A boy who has come for 2 months project work. So he led to some very significant discovery, which is being further. This is the one. You can see those things moving, no? That is the bacteria that are moving inside the yeast cell. When you see it for the first time, it was really great. Okay. So then we have done, this is the shotgun metagenome sequencing of yeast. So from which you can know growing all the bacteria may or may not be possible. But if you do the metagenome analysis, you get all the genes that are there. And then, you know, most of it, of course, eukaryotic gene because it's a eukaryote. But you also see bacteria and archaeal genes. So these are the various things that are present. And based on this, we can characterize the metagenome assembled genomes. And we gave names also to those bacteria based on this MAGs or metagenome assembled genomes. And this is the commercial growing microorganisms is a great business opportunity, and people are ready to buy bacteria. You may think I have isolated only bacillus, but I have sold bacterial cultures, and you can also do that. And I mean, by selling bacteria, we are not reducing whatever is there with us. We keep on growing and then giving to any number of people that you want. So these are the patents. This is the red pigment. This is the green pigment. And then this dietary supplement for reducing this. And this is the new one that we have submitted. This is Zushicella marina, is a bacterium which is able to produce a number of proteoglycans. This is an antimicrobial compound, and it is having broad spectrum activity against several bacteria. So this can have an application in swimming pools where all bacteria have to be killed. Initially, somebody asked for us to be used in aquaculture ponds. But in aquaculture ponds, you cannot use this because aquaculture ponds use bacteria as probiotics and for taking care of disease and all. So in swimming pools, you can use that. These are the various things that have been developed and then transferred to the industry. These are the commercialized, of course. This is our culture room, the most beautiful part of our lab with all colored bacteria. This is the group, some of them, not all of them. These are the various funding agencies from which had been getting continuously funding. They say when you get funding from government agencies, it is not great because from ours is a state government university, so you are getting from one government to another government, but you have to get funding from industry. But yes, now I am having funding from industry. Though industry was asking since quite some time, I didn't take up, but now I am taking industry things. So these, of course, this is my husband. We work together. And Professor Imhoff was the authority on photosynthetic bacteria. So we had an Indo-German project to work on that. Dr. Shivaji from CCMB, now he is with LV Prasad Eye Institute. We had gone to him just for 16S rRNA gene sequencing, which was a great thing then. But today we are doing genome sequence analysis in our lab only. And he is COVAX, with which we had a work on hydrogen production, Busse. So he is an authority on amino acid analysis. So when we didn't have the expertise, when we didn't have the facilities, just with one email, when we proved that it is a new organism, but we don't have this capability, he asked us to send the culture, and he did the analysis, and we have a common publication in that. And he's Takaichi, who has helped us with carotenoid production, and he is also from the same group. So with Vietnam also we have, but that is not there here. So whatever preliminary work that you can do in your college, you please do it. If you are a hostler, you can work even after if your university allows, you can work even after your college hours, and then have, especially because this is a microbiology group, microorganisms are a big wealth. Probiotics can be sold for 2 crore rupees a culture. That is the potential that is there. And for that, what you do, all the probiotics, there are Bacillus paranthracis, Bacillus subtilis. You have to do a number of experiments to prove if it has so-and-so capability, if it doesn't have so-and-so, if it doesn't have any antibiotic resistance, it should not have multiple antibiotic resistance, it should not have toxigenic genes. You can do the 16S RNA, you can do the shotgun analysis where whole genome sequence can come, and you can do these things. So if you are hitting something good, you can make money, you can start your own company. It doesn't really take you much time to isolate crude oil degrading bacteria. You have to only select a particular environment where crude oil is present, and then isolate bacteria, make a consortium, and then grow. And you can sell this consortium to places. There are now industries, microbial industries, which sell bacteria for wastewater treatment, bacteria for, of course, biofertilizers, biopesticides. There is so much of importance being given for organic farming, because pesticides, we have realized that pesticides are not good for our health. So these are the, whenever you go for sample collection, you enjoy, you see and all that. But there are also risks like this. It is Manali Srinagar highway, world's highest motorable road. We went and our car like this stuck in the ice. On the top, it looks like there is solid ice. But then below that, it will be thin layer of ice, and below there is water, and you will be stuck here. You don't know what to do then. Thank you for the patient hearing.
Thank you so much, ma'am, to enlighten all of us about the importance of microorganisms, their wealth, creation. And one more thing, in our microbial systematics, we just try to understand the importance of purple sulfur bacteria, anaerobic bacteria, their classification and all these things. First time I realized that how they are important for all of us. So now the floor is open for the question answers. Or rather than I say it, doubt clearance, please. Anyone else?
One request is, let it be only the question. No, before that. Let it be directly the question. Okay? Yes.
Thank you, ma'am. It was really wonderful and fascinating presentation. My question is pertain to a bioconcrete production that you have talked about. I have a question regarding how can we deal with the contamination? Is there any chance of contamination while we produce that? Or second,
Produce what? Biocolorant.
Bioconcrete using concrete using organisms.
Okay.
So is there
Okay.
any chance of contamination? And second thing is that how can we improve the strength of that particular material? Because I was working little in my lab on that, but then we stopped that project because we were stuck for that strength issues. So for bioconcrete production.
The thing here is you have to make sure that whatever culture you are going to use for bioconcrete is not a known pathogen. So once you identify the bacterium and it is Bacillus subtilis, it is likely not to be contaminant. That's all. That too, when you are identifying, taxonomic identification for taxonomic characterization, what we do is large. But when we are just trying to identify something, we'll do only 16S rRNA gene sequence analysis. And we just take a small thumb rule saying that if it is 98 point or 99% sequence similarity in 16S rRNA gene sequencing, it is the same species. If it is less than that, it is a different species. But taxonomic characterization is not like that. Even with 100% sequence similarity, we have described new species. So this is always approximation. And then E. coli is a commensal, but certain strains of E. coli can be pathogenic, and certain strains of E. coli are not pathogenic when we are fine. When our immunity is down, it may become a pathogen. So this question mark is there. But when you are doing, you have to identify the bacterium, and then you say it is generally not this thing. Of course, if you have more this thing, you can do genome sequence analysis. And from genome, you can know whether there are any genes for infectivity, genes for toxigenic genes. So those things you can also do it. And about contamination, you are telling how to analyze the strength of concrete, or?
Ma'am, how can we improve the strength of the concrete? Yeah, analyze and improve both. And second, what would be the chance of contamination? Because we are using microorganisms for constructing that concrete. So is there any chance of contamination or something like that? Or if it is so, how can we deal with it?
See, bacteria are present everywhere. It is in the air that we are respiring, it is there. In cement, it is there. In concrete, it is there already. Even without we adding, it will always be there. And when we are adding some bacteria, which are not harmful, so that is increasing the strength by forming these calcium carbonate crystals. And then they have shear strength, something, and all civil engineers have many experiments that the tests that they do, and then they say whether it has increased or not. That part I have not done. It was done in a civil engineering lab only. And the same thing can be used actually even to strengthen the thing. Contamination is, see our, what is the amount of bacteria that are present on our body? 10 times more microbial cells are there than human cells that are there in our body, human microbiome. And what we think it has come only from our parents, from genes. But the bacterial genes are contributing so much. Otherwise, if you see down south, we are rice eaters. Those people who do hard work on the road building and all, what do they eat? Rice and probably a little bit of pickle, not any fruits, not any dry fruits, not any lot of vegetables, nothing. How are they healthy? From where are they getting all the vitamins that we say are required? They are getting from their intestine. So bacteria are good. Just because bacteria are there in the concrete doesn't make concrete contaminate. I mean, it is, what is the meaning of contamination? We have purposefully added bacteria there, and we have to be reasonably sure that it is not going to cause any disease. And for specific people, it may, that we cannot say, like allergy. It may be there, but these are not like fungal spores. And the strength is coming because of calcium carbonate only. That's what the literature says. And testing has to be done in the lab. And then this crack healing is very, very important, especially for archaeological buildings, which are several hundreds of years old. There, if you have to repair it and you want to preserve your culture, then that is the best way of doing. And today there is lot of importance given for microbial processes for everything and nature-based solutions. That is the buzzword that is being used. That's what microbiologists can do here.
Thank you, ma'am. Thank you.
Directly to the question, madam.
Madam, you told about developing certain bacteria with specific features like zinc solubilization, phosphate solubilization, and to use them commercially. Now, ma'am, for that, do we need any licensing or is there any government structure that needs to be followed or anything like that?
Okay, I can tell something. As a member of expert committee on access and benefit sharing of National Biodiversity Authority, whatever biodiversity we are using commercially, we have to write to National Biodiversity Authority. It is an online form is there. You have to fill, and then they will permit you by taking. We have to provide a certain amount of percentage of on the sale value. 2% sale value, 0.2%, depending on different categories, it will be there. We have to pay after sale, we'll pay that and we get it. But mostly nobody is following this. So much is being used. Now rules are being changed. Like if it is traditional Ayurvedic medicines, there is no need like that. Different things are coming up now. But that is there. And for biocontrol agents, they have to get certification from some authorities there. For biofertilizer, nothing. And it is just a plating experiment. Just plate and see for a zone of clearing around the colony. Bus. And identify the bacterium, of course, to make sure that it is not a plant pathogen, it is not a human pathogen. Then you can use it.
Yes. Hello, madam. I have two questions on this. Generally for food, as madam has asked about the contaminants, usually in environmental isolates, when we check the application part, is it needed that we need to check like grass status or so? Like we check for the food applicants or food application of the microbes?
What is that?
Grass status, like generally considered as a safe microbes.
Yes, that is required. Generally considered, that is required. Even for probiotic use, that is required.
So similarly, when we do the environmental application of any microbes, is there any mandatory or is there any some regulations are there that by checking these parameters, we can use the particular organism for environmental application?
I'm not really sure about it because as far as I'm concerned, I gave the cultures and they did the commercialization and all, only the company has done. Okay. But because the cultures that I've given are photosynthetic bacteria, they are never known to be pathogenic at all. So they come under that generally considered as non-pathogenic, non-toxigenic. Photosynthetic bacteria are totally safe organisms. Other organisms I'm not this thing about. And for probiotics, there are a lot of experiments that need to be done as far as probiotic property is concerned and also safety is concerned. So you have to do tests in the lab. You also have to have the genome sequence. And from the genome sequence, you have to see if there is antibiotic resistance. For 1 or 2 antibiotics, it's not a problem. Multi-drug resistance should not be there. It should not be a toxigenic organism. So that is there. There are specific regulations are there.
Yeah, actually, sir, madam, question is due to the same aspect because whenever certain genera are revised, some of the texts are revised. If it is not in a regulatory list, it will be disqualified from the same level.
No, formerly, before like even Bacillus coagulans is no more Bacillus coagulans. So I had this, I had an industry project, and my student sequenced that, and he told they are telling it is Bacillus coagulans, but it is not Bacillus coagulans, it is Priestia coagulans. I told Bacillus is reclassified. Certain species of Bacillus are reclassified as Priestia, so it is that only, I told. So these things we have to be continuously seeing. What happened? Actinobacteria. What is the new name of Actinobacteria phylum? Okay. Any questions from the students?
Ma'am, I have one more question about the indicator species. Is there any defined organisms which can be considered as an indicator for the healthy environment, or which can be considered as a contaminant environment of the soil water line?
Yes, there are indicators, bioindicators. Of course, sewage contamination, we all know E. coli. And there are these diatoms, which are indicators of water pollution. Diatoms are never found in very contaminated water bodies. Like that, you have different organisms, lichens for air pollution. You have them. You have.
Okay, some more questions from anyone? Now, I request Dr. Hitat Bhatt to felicitate Shashikala ma'am. Okay, students. After that, there is a tea break of 10 minutes. And again, we will gather here for the next session, which is on AMR by Professor Arun Kharath. It's just 10 minutes as per the clock. Okay? Not by the human, as per the clock. Thank you.
phages in 1998 in Cold Spring Harbor Laboratory, New York, USA. He has published 34 research papers in international journals of repute. Today, Professor Kharat is going to talk on impact of AMR on microbiota. I request sir to kindly come on the stage for a talk and enlighten participants with your knowledge. Please, sir.
Hello. Hello. Okay, center slide. Okay. So good afternoon to one and all. Am I audible on the last? Okay. First of all, I have a question. What am I going to talk? Because since the first session, we are talking about AMR. So am I going to confuse you people? Am I going to resolve some queries? Or am I just going to bore you because you already know it? So all these three questions are there in my mind. But anyways, I have traveled all the way to deliver a lecture, so I have to deliver a lecture. So let's do that, get on a business, okay. So what I'm going to talk is basically talk the impact of antimicrobial resistance on environmental and public health. And environment already is a part of a public health, but isolate it also separately, and the human health can be considered as a second part. Initial part, I'll be talking on the human health, and then towards the other half, I will come to the environmental aspect that we deal. So here are like classes of different antibiotics used. Particularly antibacterial are not called antimicrobial, rather they are antibiotics. They can target cell wall of a bacterium. They may target cell membrane such as a polymyxin A, B, colistin nowadays. Then they could within the cell target the protein synthesis machinery, 30S ribosome subunit, tetracycline, for example, 50S subunit, the streptomycin, for example, or kanamycin. Then the biochemical pathway, dihydrofolate synthase and tetrahydrofolate synthase, dihydrofolate by the sulfonamides, sulfa drugs, and tetrahydrofolate synthase is basically targeted by the trimethoprim. DNA synthesis or DNA topology particularly is targeted by the quinolones, especially gyrates. Those are meant to maintain a DNA topology, and RNA polymerase rifamycin, rifampicin that we all know and used against Mycobacterium tuberculosis. So not that every antibiotic can be used. The drug of choice remains the cell wall synthesis inhibitors, beta-lactams, simply because humans don't have cell wall. So wondering about what would be the side effects, because some line came like treating yeast is difficult because yeast is a eukaryote. If anything you target against a yeast, likely be the targeting the human also to some extent. That's why in yeast also the cell wall targeted, because that is again a separate target for a pathogen rather than affecting the cellular system. Anyways, none. These are the different, and I would be talking largely about beta-lactams because we work on the beta-lactams. And there are 4 types of beta-lactam. Penicillin is a 1 class. Cephalosporin is a 2nd class, a 3rd class. Second is a carbapenem. And the 4th class is monobactam. So within beta-lactam also you have 4 type of pharmacophores. And also there is a non-beta-lactam peptide antibiotic, bacitracin, that is a peptide antibiotic, colistin, polymyxin. These are all the, gramicidin was the first isolated from the bacillus, polymyxin, gramicidin for the another species of the bacillus only. So the bacillus is also known to produce antibiotic. It is true that streptomyces has the highest number. World's 75% antibiotics are produced by the only one genus, Streptomyces. So there's no surprise about that. who was a chemist, not a microbiologist, right? And Alexander Fleming was MD fellow. So Chen went into the Ernst's lab, and that's where the Flore met, and both of them found. And there are still interesting points about penicillin, the world's first crystallized antibiotic. And as even today, after 96 years of penicillin discovery, it is the only most unstable antibiotic in a frozen form also. I mean, we have hundreds of antibiotics, but there is no any other vulnerable antibiotic as penicillin was, brown colored powder. And this is all is a discovery of Chen and the Flore. Anyways, but then in the Nobel Prize, he said he implicated a theory in 1945 that he said, what if this antibiotic is given at a low doses, and rather than a killing, it would favor the growth of resistant bugs, and that resistant bug will pass it on to the other naive organism. All the thing you're talking about horizontal gene transfer, didn't he speak about it? And what would happen to the suboptimal concentration of antimicrobial therapy is developing a resistance. So the theory today, couple of theories are there behind the resistant development. One of the theory was implicated by himself in the Nobel Prize lecture. That I generally speak in other lecture. I'm not going to talk about that today. But interestingly, 1942 penicillin was applied in the Second World War as a spray. Deodorant जैसे अभी spray वाला use करते हैं ना, hanky spray, just like that on the wounds of the soldier. There is also an interesting line when he visited along with Professor Wright to the soldiers in the First World War. He felt so sad because there was no medicine to treat the wounds. And luckily God had some plans, and he went with the spray, and that spray made a miraculous effect onto the soldier's wound, though not in the First World War, in the Second World War, right? But then the first report came 1943. Within 1 year. So it is not that resistance is today's business. Resistance has been there since it is applied. Within 1 year, you saw the resistance, and with the streptomycin. What is peculiar about streptomycin? There is no antibiotic word when in 1942 penicillin was used as an antibiotic. After the discovery of streptomycin, the second antibiotic of the world, from the Streptomyces genus, Selman Waksman coined the term antibiotic, and then the application of antibiotic came in 1947, but within less than 1 year, the resistance developed. Interestingly, streptomycin binds to the 50th subunit, meant not to generate a resistance so quicker. So you can imagine the kind of society, kind of exposure, kind of antibiotic concentrations that might be used is so low, but still in less than 1 year, you have a resistance. And that time there was no concept called healthcare associated infections. At that time, community associated infections were different. Those did exist, but they did not have a tag of multi-drug resistance or high infectivity, which today we have, because healthcare associated infection means hospital community acquired infections, which are difficult to deal with. Anyways, so the saga continues. No matter, bring it on, we are there to kill the antibiotic. That is continued right now, right? So here is a slide showing the different ways antibiotic resistance can be established. So let's take a look at here. Antibiotic has come in, right? And here is a plasmid. You can see that the yellow, pink, and a green color, there are 3 different mechanism encoding genes. Sufficient enough to say that many of the resistant mechanisms are plasmid borne, not necessarily chromosome borne. So now take a look at a pink product. Is basically binding antibiotic degrading enzyme. It is basically destroying the antibiotic, and that is classically happens with the beta-lactamases. Because beta-lactam antibiotic will be opening the beta-lactam ring 4-membered, and as a result of it, no more functional structure, no more activity. And now here yellow case, now that the enzyme binds to the antibiotic and modifies the antibiotic. So there are additional groups made onto the antibiotic, and modified antibiotic is not anymore active. Third class, the green one, basically produces the pores onto the membrane. The antibiotic enters and it pushes it off. That is efflux pump. So either the modifying the target site, modifying the antibiotic, destroying the antibiotic, or expelling the antibiotic, and subclass of that is also modifying the membrane morphology in such a way that antibiotic permeability is reduced. So either it is now here is this is again heterocyclic. You could see here B stands for beta lactam ring, and now it is opened by the penicillinase. Another enzymes that works is amidase, and those are the classes of beta lactams also, meaning those are not directly acting on this one, but that acts onto the amino groups. So what are the modifying enzymes? I said and antibiotic get modified. So N-acetylation, O-acetylation. We have published in molecular microbiology O-acetylation of the teichoic acid. That also results into the development of penicillin resistance. That's again strange. Penicillin resistance has nothing to do with teichoic acid, and that was the only reason this was accepted by molecular microbiology. O-nucleotidylation, O-phosphorylation, chloramphenicol acetyl transferase. CAT assay, आप लोगों ने जिन्होंने मॉल बाय पढ़ा है उनको पता होगा ना CAT assay. What is a CAT assay? Acetylation of chloramphenicol. So that is the mechanism in the organism developed, and we have utilized it for the molecular biology or expression purpose. Erythromycin esterase is there in E. coli. And also the gastrointestinal, before GI surgery, they also treat the organism's flora so that there won't be a post-operative infection. So these are some of the slides, how the resistance is developed. So you have antibiotic is broken. So the broken antibiotic is not activated, modified antibiotic, or this was a target site is changed, so antibiotic doesn't bind anymore to the target site. This is a contradict one. So these are the, see D-alanine is a normal pentapeptide, last fourth and the fifth one. Now D-alanine D-lactate, when it produces, it develops resistance to the vancomycin and the world's reserved cell wall targeting antibiotics. So you can have rRNA modifications, you can have lipopolysaccharide modification, polymyxins, particularly colistin, will be resistant by changing the lipid profile of the lipopolysaccharide, that's specifically in the gram-negative organisms. As we know, E. coli breeds within less than half an hour, so it has more ability to replicate, propagate, and pass it on to the thing. So there are like sexual mechanisms, and initially it was meant to be within the species, but nowadays horizontal gene transfer exchange can happen in unrelated organisms too. And it is not just by means of a factor. You have a transposon integrons which are also involved in the horizontal gene transfer. So now recently we have one of the article that is supposed to be coming up very soon in International Journal of Antimicrobial Agents. Here we have studied 10 years data from India, from the 5 different regions, North, South, West, Central, and Northeast region. And analysis is carried out how bad the drug resistance has happened during 2000 to 2015 and 2016 to 2020. So originally 777 papers were there. By selection criteria, we have chosen finally 89 papers. Those have a full-length article with all the proper experimental details, AST, MIC, the PCR data is also required. All the complete set of results, not just a report, are considered for the further analysis. So here is like, you can see the distribution of blue color shows northern, then the central, then you have western, then you have a southern, eastern, and northeastern. And according to that color, you would see Acinetobacter is more common. More than half of the isolates in the northern region are coming from the Acinetobacter. While if you come to the southern part, almost close to half are coming from the Pseudomonas aeruginosa. So there is a dramatic difference, right? So both of them are aerobic pathogens, but they don't behave the same way. Neither they cause the similar type of infections at the infectivity level. While you come to the west part, almost 100% infections are coming from the Acinetobacter, and it is probably North India has a higher number of publications. While central part did not have as much amount of publication. And think about central also has UP, which is 1/6 population of India. So you have to understand Indian reporting is very, very bad. not a week, it is a very bad. Hardly we report anything. And we treat antibiotics without having any idea what pathogen we are treating. And such a treatment is done at least around 98% patients. Right. So here is a distribution of escape pathogen. We were only interested in the escape pathogen. So you could see that highest number is Acinetobacter baumannii because of North India and West India, of course. Then the second comes Pseudomonas aeruginosa, third is a Klebsiella pneumoniae, fourth is Staphylococcus aureus, and list was the Enterobacter species, and second list was Enterococcus faecium, that is specifically in the wounds or more into the burn ward patients. Okay, now when we take a look at the cell wall synthesis inhibitor within India, you could see the blue color is 2010 to 2015, and orange color represents 2016 to 2020. So there is some amount of increments into the resistance for almost all beta-lactam antibiotic if you consider India as a one unit. Right, and the present paper only will talk about India as a one unit. This regional, we will be submitting it somewhere and we'll be publishing it elsewhere again. While we talk about North India, the scenario goes differently. As I told you, there were very less report from the West India. So much progressive states of West India includes Maharashtra and Gujarat, and there the list reported, right? So you could see that there is hardly any reports coming from, even East India, you have hardly any reports coming from, because that is why you don't have so much of data. However, we have sufficient data coming from the North India and South India where we can actually have something kind of a real picture, right? So it is pertinent that everywhere, almost cefotaxime, if we exclude, in all the beta-lactams, there is an increasing trend of resistance in the second half of the 2010 to 2020 decade, right? And now when we talk about the protein synthesis inhibitor, the ciprofloxacin, ma'am already told, but ciprofloxacin often is not given as a commonly, unless certainly known symptoms, such as a dysentery, for example, you have O2 or notice, where one is ciprofloxacin and one is antiparasitic. So both things are there, but otherwise it is generally drug of choice is a beta-lactam. But still in hospital, kanamycin is commonly used. Then when you have a ear infection, or you have upper respiratory tract infections, you have azithromycin, erythromycin, they are the protein synthesis inhibitors. So it's that somewhere or other where you also have a commonly used them also. So amikacin and gentamicin were used. Gentamicin is commonly for the ear and the eye infection. As a simple drop solution, you get it. So you could see in both cases also there is an increasing trend to the resistance in the second half of it. And ciprofloxacin was also raised from the 16% more resistant than that of the 2010 to 2015. Now, in India whole, and when it comes to these two regions where you have a sufficient amount of particles, yeah, that reflects that 2010 to 15 there were reports, and the later report shows more of the resistance to the amikacin, to the gentamicin, and ciprofloxacin. Now, ciprofloxacin is a gyrase A and topoisomerase 1, topoisomerase 4 targeting enzyme. While the same was there for the East India also, and to some extent, there was a reduction in some amount of resistance in the case of West India, but there were only 2 papers. So obviously, that is not a sufficient enough to conclude it as it is. Now, that was about the region-wise, but we are talking about escape pathogens. So let's take a look at the organism-wise. So now, Enterococcus faecium and Staphylococcus aureus. You could see there is also increasing resistance trend for ampicillin, for augmentin, and for Septagidium tazobactam. So augmentin is again amoxicillin clavulanate combination, right? Amoxicillin clavulanate. Amoxicillin has 2, ampicillin and cloxacillin. So it is a 2 beta-lactam and 1 beta-lactamase inhibitor, clavulanic acid. While ampicillin is a plain beta-lactam, second generation, and in both the cases you could see there is a resistance. So probably augmentin has been, there were no reports before augmentin, but now recently augmentin reports have started to come up. And same is true, even if you talk about amoxicillin, here is a meropenem and EDTA, that is a carbapenem, one of the carbapenem, and EDTA, there is also resistance increase. Again, TPZ resistance is there, augmentin resistance is there. In all, the same antibiotic profile is repeated in Staphylococcus aureus also. While in the case of Klebsiella pneumoniae, you would see the spectrum against Klebsiella, against Acinetobacter, and against Pseudomonas is increased a lot. And in these infection, often ampicillin doesn't make a choice of giving also, and they generally don't give also. So you have a ceftazidime, cefotaxime, cefepime, which is a 4th generation cephalosporin, 1st or 3rd generation, imipenem, plain, then meropenem, plain, then azithromycin, that is monobactam. And everywhere you could see there is a great amount of increase into the resistance from the 2016 to 2020. So we are experiencing more of the resistance, and probably it is because people are doing cultures. 2010 to 2015, many cases remained unnoticed because there was no culturing probability done. But then research is increased, or the hospitals are also doing more about culturing and reporting. That could be the reason we are able to get a clear picture. Same is the picture in the case of Acinetobacter and Pseudomonas. Only difference is that Klebsiella, as we know, that it is a facultative anaerobe, while Acinetobacter and Pseudomonas are the aerobic pathogens. Now, Enterobacter species, there were only reports coming from the second half of the decade, and for cefepime, 4th generation, imipenem, meropenem, then ceftazidime and tazobactam combination, and amoxicillin clavulanate. Tazobactam is also another beta-lactamase inhibitor. So even in the protein synthesis inhibitor, DNA synthesis inhibitor, we could see that there is an increased resistance for the Enterococcus faecalis, for the Staphylococcus aureus, for Klebsiella pneumoniae, in the case of Acinetobacter, Pseudomonas aeruginosa, and miraculously in Enterobacter, there were only reports coming from the second half. So we could see that there is also almost 90% or 90 to 100%, 95 to 100% resistance in the isolates is found. So this is one of the upcoming most notorious pathogen, Enterobacter. Otherwise, it was very well globally known as Klebsiella, but now in India, Acinetobacter and Enterobacter and Pseudomonas is becoming very popular. So for the beta-lactams, resistance mechanism based on the extended spectrum beta-lactamase quality. There are 4 classes: class A, class B, class C, and class D. Class A are the serine proteases, beta-lactamase enzymes that has a serine hydrolase kind of activity. While the class B are the metallo-beta-lactamases. The classical known example is a new metallo beta-lactamase. Probably ma'am was talking about it. Even I had a same feeling in 2010 when the first case was reported. It was reported in UK. And the name is NDM. And I was like, अरे यार कुछ भी हो जाए हमारे भारत को क्यों बदनाम करते रहते हो? It's not like that, ma'am. The origin of that trait is traced back. आपको बीटा डेरिवेटिव याद है? किसका बीटा डेरिवेटिव? हाँ? बीटा डेरिवेटिव नहीं याद है? ऐसे ही मेरे स्टूडेंट भी कंफ्यूज होते क्योंकि सर एक पढ़ाते समय कोई तीसरी चीज पूछ लेते हैं. कोविड का बोल रहा हूं मैं. ठीक है, कोविड का बीटा डेरिवेटिव ट्रेस बैक कहां हो गया? राजस्थान में, राइट? It appeared in the second wave, but the origin was traced into the October 2020, and second wave was in March, April 2021. So when it happened, that is outbreak. Origin is always backward. So like that, NDM was origin back to India, and that is New Delhi metallo-beta-lactamases utilize zinc, and as we all know, zinc protein. आपने सुना होगा zinc motif, zinc finger motif सुना होगा ना? These proteins have that kind of activity in active conformation. They need a metal, but if you have EDTA, it will chelate the metal ions, as a result of which enzyme becomes inactive and antibiotic gets a chance to act. So that's the second class. Third is the enzyme is called cephalosporinase. So generation 1, generation 2, generation 3, and even a generation 4, all cephalosporins will be degraded by that. Simply because it targets the main cephalosporin ring, not the side ring of the cephalosporin. Like beta-lactam is a side ring, not a thiazolidine ring. In this case, it is a cephalosporin hexameric quinoline ring is there. That ring is going to be targeted, and that's why regardless generation 1, 2, 3, 4, it becomes a substrate. And the last is a oxacelinase, which basically is a more dynamic. It acts against all kind of 4 beta-lactam antibiotic. So class A, class C, and class D, all of them combined was found to be increasing by around 7 to 8%. Looks like a marginal, but it's a considerable, right? And however, the class B enzyme did not seem to increase significantly because both of them had more or less same amount of resistance. When it comes to North India, the class ABC was increased, while the class B was reduced. In the South India, class B was increased during the period, and in the South India, class ACD was reduced. In case of East India, there was only class ACD reported in the second half, and in West India, whatever was reported was in the first half and not into the second half. About the class A and class B, both of them were like 8% only, not so much high. Now, within the organism, you could see that in Klebsiella, there was marginal increase. In Acinetobacter, there was no increase. Rather, probably there were no reports about it. In the case of Pseudomonas aeruginosa, there was increase in a class A, class C, and class D ESBLs, where the class B was marginally increased, not a big increase in this case of Pseudomonas aeruginosa. So there are enzymes, as we talked, extended spectrum beta-lactamases, and there are genes encoding for that. So those are generally called BLA to begin with. Beta-lactamase means BLA, and the remaining 3 letters are the specific for the different genes: SHV, TEM, then CTXM. BLA, SHV, BLA, TEM are the serine proteases. CTXM is for the third, that is cephalosporinase. BLA oxa is for the 4th class, D class. WIM, IMP, and NDM. These are all the metallobeta-lactamase specific class B. So class A, class C, class D, and the remaining 3 are the class C. And you could see that in everywhere, there was increase in India during the second half of the decade. And that was consistently seen in the case of North India also, especially the WIM and IMP was increased to that of the first 5 years report. So there was a considerable increase into the BLA-VIM and BLA-IM in the case of North India. Then in the case of South India, you could see there was a decrease into the resistance by the WIM and by the IM. However, the rest of the class A, the class C, the class D was increased in the second half only. In the case of Klebsiella, the resistance was increased by the class A, then the class C also from 69 to 91%. That is like a significant 28% increase is there. Marginal increase was there for the oxacillinase, but recently in India, oxacillinase are becoming more popular. Yeah, and Acinetobacter also same. You could see that in second half, you could see 83% of the isolates are oxacillinase, and oxacillinase is more dangerous because it covers all 4 types of beta-lactams. And in the Pseudomonas also, you could see oxacillinase are almost around 30% increase into the Indian population. So there is another paper being submitted to the same journal, International Journal of Antimicrobial Agents, and this is about novel beta-lactam and beta-lactamase inhibitor combinations, which shows limited activity against Pseudomonas aeruginosa isolate due to conundrum of diverse resistant mechanisms. Okay, this is under a review right now. So basically, coming back to the couple of terminologies, class A, extended spectrum beta-lactam hydrolyzing penicillin, cephalosporin, and monobactam. Class B, specifically the carbapenems and some of the cephalosporin penicillins, but not the monobactam. Class C, they have Pseudomonas particularly have a chromosomal AMC. In intrinsic resistance is already there, but it will target penicillin, monobactam, cephalosporin, but they do not target carbapenems. And class D, carbapenem, penicillin, cephalosporin, monobactam. So that is the most dangerous particularly. Now in India, almost 80% enterobacterial isolates express class A and class C type of it, and 20 to 30% express such as a class D and some of the class B. We already have seen it, around 30% of the oxacillinase have been reported in the decade of 2010 to 2020. Organisms expressing these extended spectrum beta-lactamase are representing resistance to quinolones, aminoglycosides, and tetracycline. That is even more difficult because once you have a resistance to beta-lactams, DNA synthesis inhibitor, protein synthesis inhibitor, it becomes almost no possibility left out to treat the patient besides combining more antibiotic. And combining more antibiotic is adding a stress into the detoxification mechanism within the body, right? So what is beta-lactamase inhibitor? They are generally known to restore the partner beta-lactam activity by hydrolyzing the beta-lactamase enzyme. Old inhibitors include clavulanic acid, tazobactam, and sulbactam. Now, clavulanic acid, we know clavum or augmentin. Clavin is syrup given to the children, particularly, and augmentin is given to the adults. But both of them have the same composition. Tazobactam, piperacillin, and tazobactam, or ceftolozane and tazobactam, these are all injectable, given only to the patient when they are hospitalized through the saline. Sulbactam, ampicillin sulbactam, sulticin tablet is available, and adults can take it. That is outpatient prescription. So these orange and the gray are the outpatient, while this sky blue is inpatient treatment. Now, avibactam is a one of the novel beta-lactamase inhibitor, and it is combined with the ceftazidime avibactam. In 2016 onward, Pfizer has produced, and USA started utilizing. We published one paper in 2019 that more than 700 isolates we had, and nearly 500 isolates did have resistance to ceftazidime avibactam. And ironically, there was no approval to ceftazidime avibactam to be used in India. It has been given only to the COVID patient who are on ventilator during second wave of the COVID. So you can imagine, we have published ceftazidime avibactam resistance before, 1 year before, it was approved only to those vulnerable population about to die, right? So that in any case, we are not going to talk, but this boy has used, Prashant has used more than avibactam in this paper. I'll talk about what did we use in ceftazidime avibactam, then ceftolozane and tazobactam, which is existing composition, and one more, imipenem and relebactam. So we have relebactam and avibactam as a new novel beta-lactamase inhibitor. How these two are different than the tazobactam or clavulanic acid or sulbactam? The fact that those don't get recycled. So these avibactam and relebactam, because they have high turnover rate and they can be recycled, they don't get metabolized, you can get away with the lower concentration of them. Because other traditional 3 beta-lactamase inhibitors, they don't have a high turnover rate. You need to provide them again and again with high doses. So here, with a low doses avibactam or relebactam, will be able to help ceftazidime and imipenem respectively. Ceftazidime, 3rd generation cephalosporin, imipenem, carbapenem class of antibiotic. Now, 16 tertiary care hospitals we have taken in study, and there are more than 400 isolates being taken care of. Okay, samples were also collected during before the COVID-19, so there is no mistake. And as on today, meaning ceftazidime avibactam was not approved, imipenem relebactam, even today it is not approved to be used in India. Okay, and this is only for the research level, and we are worked on it. So none of the studied beta-lactamase inhibitor were approved to treat in Indian cases. Resistance being reported is among naive isolate. These haven't been exposed to the clinical setup as normally most of the antibiotics resistance we can imagine. So from second wave of COVID-19 to the inpatient, Cas-IV, that is ceftazidime and avibactam combination, has been approved by the government of India. That's an emergency approval. So what we see here, cefepime, 4th generation cephalosporin, ceftazidime with avibactam, then ceftolozane with tazobactam, piperacillin with tazobactam, imipenem with relebactam. We have seen that nearly more than 52% isolates that we have isolated exhibit resistance to ceftazidime avibactam, ceftolozane, tazobactam, as well as imipenem relebactam. So which is very surprising that despite having no exposure, they have a resistance. That again says, without a reason, during horizontal transfer, genes may have transferred in these organisms that we did not know what all they have acquired, right? And so then in vitro activity was done for the amycin. You could see that almost resistant bugs for the ceftazidime, 85%. Combination again remain the same, 85%. Even with the avibactam, then ceftolozane, tazobactam, 88%. piperacillin tazobactam 84%, imipenem 100%, which comes down only to the 80% with the relebactam. So that's a surprising. Still, you have 80% of the isolates being resistant to the combination, which is still not known to be used in India, not yet in the market. So, and meropenem, that obviously is a plain meropenem, will have a very high resistance. So, based on the MIC of this septazodim iobactam against imipenem, these are also again further classified. So you could say that more or less this study population showed there was a resistance to imipenem, septazodim, ciprofloxacin, amikacin, colistin. Colistin is a lipopolysaccharide targeting antibiotic, not a beta-lactam. Now 42%, 44%, 54%, 50%, and 20% respectively resistance is there, very high. Susceptibility rate to the ceftolozone, avibactam and relebactam combination was 55%, 60% and 62%, which is again concerning as far as the availability of antibiotic is considered. Genome sequences revealed that mutations in PBP3, in addition to OPRD, that is going to control the efflux pump or permeability membrane, cause a resistance, but resistance to fosfomycin, polymyxin, quinolones, aminoglycosides. So therefore, these organisms are not just MDR, but they are XDR and PDR. XDR are those where the MIC is very high, and PDR and XDR are only differs by their MIC amount. If you have a resistance to all prescribable antibiotic, it is called PDR. But if you have high MIC among the PDR, it is called XDR. Right. So among carbapenem resistant Pseudomonas aeruginosa isolate, NDM was the most common in India. Right. And in the non-metallo-beta-lactamase, meaning there is no NDM, there is no VIM, there is no IMP. In this case, there were like other mechanisms we have identified, and this is what we are discussing in this paper. So coming to the second half, environmental health, as you could see, you've seen in the morning, the soil health, eco health, human health, plant health. So here you can see aquaculture. This is the source of water. Think this as a Ram Teri Ganga Meli wali nadi hai. Pahadon se behti aa rahi hai. And this water is being used for drinking. This water goes to the agriculture. So you could see irrigation of the water is going. Then the water is used for the domestic purpose. Water is used as a food also to the human beings. So human are using, agriculture is using, agricultural harvest is coming. Then what is happening? Aquaculture, water sewage gets into the treatment plant. Then humans, hospitals, directly sewer lines or hospital sewer lines goes to the treatment plant. So aquaculture directly through the human also comes again to the treatment plant. So aquaculture and farm runoff goes to the aquatic sources. So aquatic sources are getting contaminated with the materials which are present into the litter, the aquaculture, and litter is same is used as a manure is used for the agricultural. That also is contaminating into the vegetables, fruits, etc. So eventually we are having a mess or a soup, and in the soup you don't know where is sabji, where is tomato, where is potato, and where is starch. You just spill it, right? You just spill it. So we are in the soup here situation, and we don't know what we are drinking, right? So actually Ram Teri Ganga Meli jo movie in 1986 ki, there was one movie before that, 1950. There was a song called ओठों पे सच्चाई रहती है, जहां दिल में सफाई रहती है, हम उस देश के वासी है जिस देश में गंगा बहती है. सेम राज कपूर 86 में बोल रहा है, राम तेरी गंगा मैली हो गई पापियों के पाप धोते धोते. What does that mean? It was not about Mandakini, it was about the river Ganga and the effluent that was thrashed into the Ganga. You think of the drop at Gomukh, the उत्तरी थी धरती पे लेके कितना पावन पानी, वो पावन पानी कहाँ? गोमुख में. जब कलकत्ता में पहुंच जाती है तो गंध बन जाती है. So you saw her everything like a songs, picturization, and you lost in that. But actually it wasn't about that. It was a very social, environmental. जितनी मेधा पाठ करके आंदोलन लगते उतनी ही तगड़ी मूवी थी वो, मगर हमने उसे मंदाकिनी को एक और राज कपूर के टिपिकल पैटर्न में बैठा के देख लिया. दैट इज रॉन्ग थिंग टू डू इट. Right, and we say against the plastic, but while going to the Yamunotri Gangotri, I have photos. You will carry at least sufficient enough of bottles with you, and you will track them without thinking who else is, and while thinking, अरे यार कैसे लोग करते हैं, इनको समझ में भी नहीं आता, अरे तुमने भी 2-4 छोड़ के आए हो वहीं पे. So that's the way we are. Like we say that शिवाजी शेजाऱ्याच्या घरी जन्मलेला पाहिजे. मतलब शिवाजी का जन्म होना है मगर पड़ोसी के घर में, अपने घर में नहीं होना चाहिए. राइट. सो योद्धा हो या नेता हो, दूसरे के घर में हो. राइट. अपने घर में नहीं होना चाहिए. सो हियर इज द होल वाटर is a source for the soil, is a source for the food, is a source for everything. And on that topic, what is happening in this water? The quantity of antibiotic, you will say, अरे sir कितनी कम आती है. Almost non-detectable. Right? Almost non-detectable with your crude methods. You need a very sophisticated method to detect whether they are there. बहुत कुछ होता है. Because things, as we know, have their effect. Arsenic बोलते ही tentacle जग जाते हैं ना. Arsenite contamination है, cancer हो जाएगा. कितना है? Not in milligram, in nanogram, but that is also killing, right? Similarly, antibiotics concentration, according to you, is too small, nano, but that is good enough for organism because that acts as a selection pressure. I was about to say, somebody had asked in the morning about antibiotic in agriculture or husbandry, right? So actually, ऐसे नहीं होता है, वहाँ नहीं है, कम है, ऐसे नहीं देखना चाहिए. Probability is there and it is going to multiply because of horizontal gene transfer. So we have to take this as a call, alarming call, and work out from now rather than tomorrow. Tomorrow तो आपके हाथ में कुछ बचेगा नहीं. You have to work at any cost because you are going to be on SOS mode at that time. But right now there is a lot you can do and think about it. So that is acting as a selection pressure, and because of selection pressure there is a development of resistance. I have 2 PhDs to talk about. One is about this and about the selection pressure and developing antibiotic. So this is again the same thing. There is a proposal like how to do the self risk assessment. You should do into the hospital, the field, the husbandry, wherever animal, poultry, etc. Stock animals are going and find out the organisms. You'll find some amount of resistance is there. And if you just open PubMed, you will find. ना बता देने से प्रॉब्लम खत्म नहीं होती है. भारत नंबर 1 पे है AMR में, no doubt about it. Just because we are number 1 population could be one reason, but we need to report so that sensitize the people. पदुकोण के advertise है, painting में antibiotics वाली है ना. Don't do that. Don't do that. Luckily, I saw one video today. One of my friend from ISRO, who is a scientist, जी, sent me a video. कोई एक बड़ा मॉल खोला, किसी एक बुजुर्ग को बुलाया इनॉग्रेशन के लिए. ये बेनेवोलेंट लगता है ना किसी को एक रैंडम व्यक्ति को बुलाना, वो भी बुजुर्ग को. एंड देन एट द एंड ऑफ द पॉइंट शॉपकीपर सेड, मालिक बोल दिया कि भाई यहाँ 21,000 प्रोडक्ट्स है, जो आपको चाहे ले जाइए. expensive brands, but he says I don't need this all. उसमें कोई, अगर मैं वीडियो आपको शेयर करूंगा, आप शेयर करना बाकी लोगों के साथ, कि उसमें ऐसी चीजें बताइए, those were not required and never were there. किसी बिल्ली को कैटरैक्ट हुआ है क्या? उसमें बुलाया. इतने सारे तुम यूज करते हो आंखों पे, स्किन पे, फिर भी आप ही को प्रॉब्लम है. I don't need all that. That's a one line message I would say. So the thing is that here is resistance, here is also resistor, and here is also resistance. So we need to understand resistance in a very proper way. And this is almost a 10-year-old story. It's not that we are talking about 2025. We are talking established fact in 2015 coming from the Europe, and that is also in Nature Review Microbiology. So I think it's a pretty old story. We are talking almost 10 years before that, because this publication had to have 10 more years before story. So right. So what happens in sub-MIC? There is a selection dynamic. I purposefully added this slide because this morning what happened, and after that I sat and I added a slide. So what happened, it basically enriches the pre-existing mutants. Though it is a low amount of antibiotic in the environment, it will enhance the selection pressure, and there is also another pathway called de novo selection, and de novo is not a horizontal. De novo is individual. जैसे हमारी family में किसी को diabetes नहीं था, फिर हो गया. That's not horizontal. By birth is horizontal, probably. Because that is also probability. You never know after fertilization how many cycles have happened and where the mutation may have taken place in that unborn child. But during prophase only it's a genetic. Rest of the thing after the first vertical division, everything is individual cell division related pressure and that pressure related mutation, right? So that is a de novo. Then second mechanism is genotype and phenotype variability. It creates a mutagenesis, it creates a horizontal gene transfer, promote the phenotypic variability. Also, intracellular signaling cycles are also changed. जैसे मैंने OPRD बोला इसमें, that basically is to do with the efflux pump regulation. So you have a quorum sensing, virulence factor and biofilm formation. So don't think that there is very little. No. जहर ज़्यादा नहीं पीते, चॉकलेट केक ज़्यादा खाते हैं, right? मगर दोनों का इफेक्ट सेम है ना, एक में प्लीजेंट रिजल्ट है और एक में कितना भी खाओ तो कम लगता है, वो थोड़ा सा भी खा लो तो मर जाओगे. It's kind of that, right? So it's a situation is that. So what Bipin has done, Bipin was working with ceftazidime at sub-MIC. So we have 1/2, 1/4, 1/8, 1/16, 1/32 of MIC concentration lower. So we wanted to see, get the antibiotic concentration that does not show the growth defect. So you could see 1/2, there is a problem, but 1/4, 1/8, 1/4 onward, none of them microbiologically call there is a problem in the growth. Growth has no problem. But then, मैं हमेशा बोलता हूँ, किसी ने कोर्ट में जाता है डिवोर्स के लिए, कहती है कि इसने मुझे चाटा मारा, उससे कोई डिवोर्स नहीं लेता है. That was come to the tolerance level. घड़ा भर गया था पहले से ही. And it was just waiting for a movement. So चाटा बड़ी बात नहीं है एक्चुअली. उसका प्रिटेक्स्ट भी देखोगे उस दिन का तो ऐसे लगेगा कोई ऐसी बड़ी बात थी ही नहीं. बट उससे पहले जो हुआ है, that she has tolerated for years, that is unbearable. And that is called a real stress. When we say I'm in stress, that means your body has already given it up. But body was already working on managing your stress without you knowing that it was a stress. And this is what 1/4, 1/8, 1/16, 1/32 is. Looks like things are fine. Everybody looks like दोनों जब बाहर निकल रहे तो made for each other लगते. भैया ऐसे नहीं है. Right? It is just a body shop. And बाकी कुछ नहीं. जब अंदर जाती है तो उसे ही पता है क्या है. Right? So this is a kind of situation. So what happens here? MIC, you will see a lot of cells are lysed. 1/2, cells have become a spherical. So there is abnormality. And then even by 1/8, you could see the elongated cell. So that is also defect into the cell division. Right? You have multinucleated cells formation. And then at 1/32, more or less cocobacilli are formed, just the way it is supposed to be for the Acinetobacter baumannii. The scanning electron microscope also was seen at no antibiotic. This is 1/32, you could see the proper diplococci. While in the case of 1/32 also and 1/16, you could see the cells are elongated. And there's a lot of things between the cells connecting. So there is some molecules extracellular matrix is being produced. Cell shape is changed, surface morphology changed. Just because I don't see the difference in the stationary phase OD and doubling time does not mean all is well, right? मैंने जो जोड़े का उदाहरण दिया ये यहाँ पे fit होता है. Okay, so we wanted to test the ability to produce a biofilm and check it out whether really. So without antibiotic, let's say here you have 1 OD. At half MIC you have slightly less than 1 OD. Then 1/4 it is gone to the 1 OD, 1/8 it is slightly increasing, 1/16 is increasing, 1/32 is increasing. So क्या हो रहा है? एंटीबायोटिक तो है नहीं, फिर भी बहुत कम है. आपके हिसाब से कहोगे तो 1/32 is very low antibiotic, but still it is feeling a stress and it is increasing the amount of exopolysaccharide believing like 7 में आ जाओ जैसे, right? 7 में आना मतलब when you are in community. राइट जैसे ही आप वैसे जोड़े की पत्नी जब मैके जाएगी तो भूल जाएगी 2 दिन में पति के साथ हूँ. शी विल बी विद द फ्रेंड्स. That's a biofilm, right? So there is a biofilm formation because there is a stress, right? So here it shows, although at this point onward, 1/4 onward, there is no visible difference in the growth parameters. But there is a lot is happening inside the cell and you have to understand that. Means agar itna sara बढ़, what is known about resistance? Resistance of a single cell when it is replicating as a suspension culture axenic, and the resistance as a member of the community in the biofilm is never same. अगर वहाँ 2 माइक्रोग्राम है सिंगल सेल का तो यहाँ 200 माइक्रोग्राम होगा. Now the question comes, when the Acinetobacter pseudomonas klebsiella produces biofilms into the patient and resistance is increased by the 100 time, you can never reach to the concentration required to kill the pathogen, because that concentration will kill animal host itself. So that is a big problem, right? So this is the issue we are talking. So what happens in that case? The drug inference, persistent cells are also produced and they will develop, persistent cell will create a mutations. So it is a very small fraction, less than 0.1%, but 10 to the power 8 ka 0.1% is also 10 to the power 5 cells. So that's a lot of cells undergoing mutation, and different mutations are going to produce. So the point here is that don't think that it is just a little and that doesn't make a difference. It makes a difference. And there are like lots of things are happening. Lipopolysaccharides are being extruded, multi-drug efflux pumps are also being developed. All those things are happening. Porins are taking place. Then another point, one of the students is submitting. This guy has submitted already thesis, Bipin. Now this is Durgesh, who is going to submit in December. I'm just giving one slide about his work. How bad this was. I'll just go back to this slide. Yeah, from here, right? This pathway. Pani, kheti, and fruits, vegetables, right? This is where I'm coming now. Yeah. Okay. So there are 4 sites. Ramganga, which is a river in Bareilly. Now there is a river taken. There is a small confluence also, vegetable plot grown on the bank of river just 200 meters away. And there also we take a free soil, we take a rhizospheric soil, and we take a phyllosphere. Kalindi Kunj in the Delhi itself, Yamuna river. So different river, different type of activities. Then river water, domestic sewer line is also meeting there, vegetable plot, free soil, then rhizospheric soil, phyllosphere of the plant. Varanasi Ganga river, again there is a river, vegetable plot, rhizosphere and phyllosphere is taken into consideration. Lucknow, Gomti river, vegetable plot, rhizosphere and phyllosphere. Why I chose Bareilly? Very simple. झुमका गिरा रे बरेली की बाजार में सुन के पागल हो गया था. So I thought like Bareilly should be there. The reason is that Bareilly is known as a Bas Bareilly. So there is a different type of effluent, right? Bas Bareilly मतलब बाम्बू का काम जो होता है वहाँ का उतना, और फिर वहाँ पे मेटल इंडस्ट्री नियर बाय है, तो मेटल का भी effluent में जाता है. झुमका गिरा रे वो lighter part है. But these are the 2 different species which are present in the river Ramganga. Then Kalindi Kunj, we know Delhi is a hub for anything and everything, and the most polluted city of the India, at least I would say. And Varanasi, we know it's a religious activities, and also the other, there are not known big industries, but religious activities are also associated. Lucknow has a cement industries and pesticide industries too many, so that is why the Gomati river. I chose all 4 different river, 4 different activities, so that what we get into that is a question. And then we chose 4 varieties, palak, methi, coriander and beetroot. Very simple, hum sab khate hain. And the summarization to tell is like, I'm being very diplomatic while putting the resistance. 60% are MDR. अब MDR बोलते पेपर में पूछोगे, ग्रांट में पूछोगे, पहला सवाल आएगा कि BSL2 फैसिलिटी है क्या? भैया यहाँ कौन सी फैसिलिटी है? ये क्या कर रहे हो? Out of them, 15% of them are producing ESBLs. And nearly 2% are producing non-beta-lactam, non-MBL beta-lactamase. That is even worst, right? So सबसे खतरनाक चीजें हम खा रहे हैं, और हम BT ब्रिंजल लेना है क्या नहीं लेना है इस पे चर्चा कर रहे हैं, right? So another topic that would directly talk about a society, and that recently we have carried out work under the ICSSR project. Knowledge and attitude towards antibiotic use and identification of financially feasible options to curb the antibiotic resistance in the environment, particularly India. We have 972 participants in this case. And there were 5 categories: senior secondary school, 11, 12, PUC, what we call. Then senior college, that is graduate, postgraduate students, their teacher, medical college student, their teacher, paramedical college, their teacher, student, learned groups from the corporate and NGOs, right? So all these different people were taken into consideration, and they were checked for the level of knowledge about AMR, use and abuse of antibiotic, and action towards the resistance. So it was found that Although most of them, like 95% of them say we have heard antibiotic, they have read from the common knowledge, books or advertisement. But you can see that they also believe, almost 75 to 80% people believe, like rural and urban, that we can differentiate. कि एक antibiotic सबके लिए नहीं काम करता. You need a separate antibiotic for separate thing. That is what people say. Then the cause of antibiotic resistance, people say largely body become resistant to antibodies, and antibodies has nothing to do with antibiotic. So that is where this was a catch question, right? सब टिक 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 करते ना लोग, तो एंटीबायोटिक एंड एंटीबॉडी का फर्क ही नहीं समझ में आना है. सो यहाँ पे पता चलता है कि हाँ, इतना जो इसने बातें बताई ना, सारी ऐसे ही बता दी है. So that means like overuse of antibiotic, only rural people said like कि हाँ, ज़्यादा यूज नहीं करना चाहिए. रूरल में वैसे भी कम यूज होता है एंटीबायोटिक्स का. Then wrong indication was also indicated more into the rural part. Then perception, antibiotic resistance affected component. इनमें कम लोगों को लगता है कि ये प्रॉब्लम इंडिया का है. Most of the people feel, 40% feel for the global problem. Indian people, इंडिया के प्रॉब्लम में मानने वाले उससे आधा है. That means वही अपना बेटा बाबू है, पड़ोसी का बदमाश है, right? Global problem है, हमारा नहीं है, right? 40% versus 20%, right? That is what, this is a perception. So these are the, when you go into the people, you really learn what people and how do they diversely think about it. Now, indiscriminate use of antibiotics में देखा, ineffective treatment, emergence of resistance, financial burden, increased antibiotic in the environment, prolonged illness. इसमें भी हम लोग देख रहे हैं कि almost 40 to 80% people had a different kind of a response. Use and abuse में, although it seems that city people, 90% and close to 90% people from the village are also taking antibiotic by the doctor, but the possibility of prescribed by the family member who knows nothing, right? Then repeating the same antibiotic, almost again 30% in both the cases. Slightly around 30 to 40% it is presumed. जैसे आप फार्मासिस्ट के पास चले जाओ, आप सिम्टम बता दो, वो देता है, that is a presumption based, right? Or then again repeating the antibiotic, even if it is not the same condition. So you could see that is very high, 65%. So there is like abuse is taking indiscriminately, just because कोई बड़ा बता रहा है, भैया बता रहे, पिताजी बता रहे, पड़ोसी बता रहे, टीचर बता रहे, कभी कभी माइक्रोबायोलॉजिस्ट को लगता है हम डॉक्टर ही है, आई एम ऑल्सो माइक्रोबायोलॉजिस्ट, राइट? तो ये सब होता रहता है. So reasons for antibiotic consumption without prescription, then what are the issues? और डॉक्टर ही नहीं मिल रहा है, पता है, उनकी फीस ज़्यादा है और मुझे डॉक्टर पे भरोसा नहीं है. That is a major. 30% in rural and 40% in city. Can you imagine? This is a high amount of population believes that fees is high, or I don't believe in the doctor, right? Second reason is I know it works, 30%, 30%. So if pull this 30-30, it becomes more than 60%. Can you imagine? I mean, wrong reasons are there for increasing the resistance. Then available at home, another 20%. So 60 plus 20, 80%. क्योंकि घर में है, क्योंकि मुझे पता है, और क्योंकि मुझे डॉक्टर पे भरोसा नहीं है. तो ये तीनों चीजें मिला के 80% हो गया. Will that sufficient? Will that attitude work out? That's a very wrong attitude. This is where we are living. And remaining 20%, friend and family advice और non-acute disease था. मतलब acute की definition भी हम करते हैं. इतनी तकलीफ नहीं थी, नॉर्मल कोल्ड हुआ था. कोल्ड तो हुआ है, फिर भी कैसा नॉर्मल हुआ था. ये कॉमन टर्मिनोलॉजी है ना. क्या हुआ, क्यों नहीं आते? अरे नॉर्मल था, खांसी जुकाम, मतलब नॉर्मल कोल्ड हुआ था, नॉर्मल फ्लू हुआ था. सो ये क्या है कि इतना कुछ नहीं था, ठीक, दवा खा ली, ठीक हो गया. So people perception about antibiotic can be repeated. For fever, people use it 70%. Common cold, again around 60 to 65%. 50% for malaria and 40% for measles. So these are the infections or the conditions that can never be treated by the bacterial antibiotics. इतना use हो रहा है. Side effects, vomiting, lack of sleep, loss of appetite, skin irritations, आपको लगते हैं, कुछ लोगों ने बताया ना. Of course, every antibiotic would have some side effects, so we cannot blame anybody or complain that he or she was just complaining like that. It could have happened with them. And now coming to the team, antimicrobial, Bipin Yadav, who has submitted a thesis. वेजिटेबल एंड इस पे काम करने वाला दुर्गेश कुमार है, ही इज सबमिटिंग इन डिसम्बर नाउ. एंटी कैंसर ऑल्सो वी वर्क, धीरज and Nilesh is working. He's a Gujarati. Swati is working on pesticide degradation. Sapna has moved now out. She is in Ohio State University. She worked on pesticides. Abhishek is a bioinformatician. Here, Nilesh and Dheeraj works with me, though they are cancer is their main forte, but they are working with me for Ganga and Yamuna. गंगा भी मैली हो गई है पहले से, अभी यमुना जी भी मैली हो गई है. उसका भी एक साल के अंदर पेपर आ जाएगा कि कितनी मैली हो गई हम बता देंगे आपको. ओके, सुडोमोनास इरोजेनस पे काम करने वाला प्रशांत है, फिर समर्पिता है. कोलैबोरेटर्स में बालाजी वीर राघवन CMC वेलोर, then Mark Fullerle from University of Rouen and Professor Corinne Mercier from UGA. The friends from various agencies are also gratefully acknowledged. I'm open for any question if you have. Thank you very much. Yes.
Thank you very much, sir.
बहुत खुश हो गए, खत्म हो गए.
It was wonderful lecture actually, sir. You have very in detail you have explained about the impact of AMR on microbiota, sir. Any questions from?
पहले बता दो, केवल एंटी नेशनल नारे नहीं लगते JNU में, ये भी देखना. मैंने जानबूझ के लाया है. That's my lab.
Sir, I have a couple of questions. May I?
Yes, please.
Yeah. So first question is that the basic understanding that I have, I may be wrong, I don't know, that beta-lactam antibiotics normally work on peptidoglycan synthesis, especially stage 2, stage 3, where the those networking or the cross-linking takes place, right? And sir, the organisms on which this has been tested, only one was gram-positive, that was Staphylococci, and the rest...
Enterococcus is also gram-positive. Enterococcus
and the rest were gram-negative organisms.
Okay, I'll answer before you go on the question.
Yeah.
Originally, Cecil Pan, before Nobel Prize, treated minor eye laceration, Streptococcus pneumoniae, and an infant born to a gonorrhea mother, gram-negative. So it is known. And it is not a gram-positive species, that's wrongly said in the classes. Penicillin or beta-lactams works against actively replicating organisms. It's a broad spectrum. Third generation cephalosporin only gram-negative. First generation against gram-positive. Second generation little more for gram-positive, less for the gram-negative. And fourth generation equal for the gram-negative, gram-positive. So I think your question, that question is answered. Pucha dusra.
Sir, second question is that as in India, I don't know about other places, in India, we do not have a very stringent procedure for the biomedical waste segregation or its disposal like that. Even health and hygiene, sanitation, all these are the problems. So sir, most of the pathogens directly get entry into the water bodies or into the soil, and there the routine organisms who are not yet multi-drug resistant or having any resistant, they get it through horizontal transfer as you have suggested. So it is like a vicious circle that is continuously going on, and we are going for the next generation antibiotics and having more side effects and all those things. So do you think the better disposal techniques, the better health and sanitation or hygiene practice can somehow affect to lower this down?
Okay, एक कहावत है ना, लातों के भूत बातों से मानते नहीं, इंडियन सोसाइटी को अप्लाई होता है. द वेरी बेस्ट एग्जांपल फॉर स्टूडेंट टू अंडरस्टैंड, कोविड में डंडे लगे तब घर से बाहर जाना बंद हो गया. तब तक लोग क्या होता है करके निकल ही रहते ना, क्यूरियोसिटी है. सो व्हाट यू सेड, एक्चुअली बायोमेडिकल वेस्ट एक्ट is passed in 1987. And there are at least half a dozen modifications and amendments have been done. During COVID, there have been 3 amendments made. So actually it is there. But for anything and everything, implementation is a bottleneck in India. See, theoretically, no antibiotic or no drugs are to be sold by the pharmacist. I mean, it is only and against prescription only. But unfortunately, you live in the democracy and people, rather than their responsibility, they believe that Article 14 has given me the right, freedom of speech and freedom of whatever. And I come from the land of freedom, JNU. So, uh, that's wrong. What you said is very true, but it is not correct to say we don't have rules in place. We have rules in place, but who will hang the bell in cat's neck? पकड़ो दो चार को डंडा कर दो, ऑटोमेटिकली एक मराठी में कहावत है, पुढच्याच ठेच मागचा शाना, मतलब अगले को ठोकर लगते 10 लोग सीधे हो जाते हैं. तो डंडे कुछ बच्चों को पड़े, बाकी अपने आप घर में बंद हो गए ना. सो कोई तो इम्प्लीमेंटेशन में जब कोई कमिश्नर आते हैं किसी एक डिस्ट्रिक्ट में, स्ट्रिक्ट होता है तो वो डिस्ट्रिक्ट राइट हो जाता है उस पीरियड के लिए, एंड जैसे ही उनके तबादला हो जाता है दे आर बैक टू द स्क्वायर. वही हुआ रिवेंज ट्रैवल और रिवेंज टूरिज्म केम इमीडिएटली आफ्टर सेकंड वेव ऑफ द कोविड. लोग थक गए थे, एक साल पक गए थे, सो दे स्टार्टेड, अरे जो भी होगा देखा जाएगा. सो दैट इज एटीट्यूड. We have rules, but for the environment, for agriculture, for prescription, we have rule, and biomedical waste. Since 1987, we have that.
Sir, last question.
Yeah.
Sir, antimicrobial agents are normally produced by microorganism as a mechanism of self-defense.
No,
The one who is producing
that
it.
is not a defense.
Okay.
That is, it's a need to make space in the niche, and others
Yes.
will make their own ways to survive there.
Exactly.
See, predator and prey relation is a blessing in the
Exactly.
eco.
So, sir, doesn't it implicate that every individual microorganism has an inbuilt mechanism to resist these antimicrobial agents?
Not necessarily antimicrobial, every kind of thing
Every
that
kind
happens
of,
in the
yes,
nature
yes.
environment, they are
Yes.
blessed to, and that's why survival of fittest works, right? Those who can sustain will survive. Those who don't sustain will eliminate.
So, means it is in fact an inbuilt mechanism.
It is. It is. But just is that when we say AMR, we are talking about their equilibrium in the nature what exists is disturbed.
Is disturbed,
because of
yes.
anthropological
Yes.
intervention and disobeying the rules regulation.
Okay.
Rest is everything. See, antibiotic producers are organisms. Antibiotic nullifier are also organisms. So they are there in the nature. But their both quantity in the ecosystem is well maintained. That is what I call an equilibrium. What COVID has done? COVID has mutated. COVID कोई जाने वाला नहीं था. Go COVID कुछ नहीं होता है. हम आए हैं तो बैठने के लिए यहाँ आए. We will establish equilibrium. We will mutate in a way. अरे भाई सारे लोग मर जाएंगे तो COVID भी नहीं रहेगा ना. So COVID had no option but to mutate, and kind of a way to figure it out, it becomes a common cold. Now it is. Are we bothered about it? So one natural system में predator prey relation in a small limit, जैसे हम बोलते हैं ना, trace elements are required in below threshold, up to a threshold. Similarly, predator prey relation is the blessing to the ecosystem to sustain. आपके BT cotton का misuse होने की वजह से honeybees को आना पड़ा, because honeybees ran away. You made them run away because refugee crop was not used. Farmer भी industrial economy का है, right? Because he didn't want to lose some space. तो ये है.
Thank you, sir.
Any more questions?
Last question from my side.
Okay. Students को समझ में आ गया इतना? Oh, I'm not a good teacher. I'm a very bad teacher.
No, you can't say like that because I am your student. Okay. So just only one question from my side. As we discussed earlier, it's the now time to rechange or reframe the definition of MIC. Because as per your presentation as 1:64, 1:32, I think it's now time to reframe the MIC.
No, we are reducing. We are trying to use a lower level than the MIC, below MIC, 1/2, 1/4.
My question is how they can determine the MIC value?
See, MIC by theoretical definition is the concentration that results in a 3 orders of magnitude reduction, theoretical definition. So if you have a 10 to the power 6 cells produces a microcolony, so you should have no zero. 10 to the power 3 is MIC, if you calculate. But we know that at 10 to the power 3, you don't get OD, you don't see the colony. So that is the way we see, like on the plate or in the broth. If you take optical density, the lowest concentration that inhibits, right? Inhibits means we don't see the growth, either in the term of spectrophotometer or on the plate. But theoretically, if you estimate a CFU, it's not a zero. It's a reduction by the 1,000 times. That's the definition. So any concentration that gives a 1,000 time reduction is MIC. Yeah. Okay.
Thank you, sir.
Okay. Thank you very much. Thank you very much.
Yes.
Okay.
She has a question later.
Hello, sir. I am working on quorum sensing and its inhibition. And by taking this AMR as a research problem, I never think about your work. It is really helpful for me to identify the research problem. So my question is, whenever looking about the quorum sensing inhibitor as a potent molecule against the, or by solving the problem of AMR, I am checking about the marine microorganisms and its its a biochemical molecule for treating the multi-drug resistance species. For study, I am taking a Pseudomonas aeruginosa. In laboratory scale, I just get the results, but I didn't check it molecular level. So please little emphasize me on a molecular level of quorum sensing inhibitory molecules with the AMR.
See, in the case of Pseudomonas, you have chosen a very good organism, in fact, because there is a wealth of information available on the
Yes, sir.
biofilm and AMR strains of Pseudomonas, particularly
Yes.
POW1, which is a very globally standardized strain. And the players are also known, meaning the elements, molecular players are also known,
Yes.
which are known to produce a biofilm, which are known to be affected with the anti-biofilm components, if I understand what you're trying to say, the same thing. So maybe you need to identify those names of the gene from the literature and just see their PCR-based, real-time PCR-based analysis. That would be the easiest way to do so.
Okay, sir.
Okay.
Thank you.
Thank you, sir. Now I request Dr. Abhijit Joshi, sir, to felicitate Professor Arun Kharath, sir. Thank you very much, sir. So with this, all 4 keynote as well as special addresses are over. Now there is a poster presentation at Yoga Hall, 3rd floor. Volunteers may guide the participants about the venue. And the small instruction for tomorrow at sharp 9 a.m. we will start oral presentations at the same place, right, this auditorium. And before we leave, please rise for the national anthem. I request audio team.